This meeting is being recorded.
May I share the screen?
Yes, you can start sharing the screen.
Yes, thank you very much.
Welcome everybody to this session about a stochastic model for neuronal activity. Let me start thanking the organizer for this opportunity. And each speaker will have 20 minutes plus 5 minutes for questioning. And our first speaker is Ryota Kobayashi from the University of Tokyo, who will talk about modeling spike frequency adaptation of a neuron and its potential role for sensory encoding. Please, Ryota, can you start?
Yes. Thanks, Laura. Thanks, and thanks for inviting me as this session. And I will talk about this title, but mainly today I will talk about the first part about how to model a single neuron or something like that, and the mathematical modeling about single neurons. And today I will talk about the information processing by a single neuron, and so maybe most of you knows about it, but I will briefly summarize what is happening in the neuron. And neuron receives the input current, and this current consists of excitatory and inhibitory, some of excitatory and inhibitory current, and they receive fluctuating and noisy current. And receiving this noisy current, our neuron is an element in the brain, generate spikes. And spikes is very important. And this spike information is conveyed the further neurons. And we are interested in how this spike information is processed in the brain, and it is to develop or consider potential application, how this scheme can be used, and so on. And briefly, in my talk, I summarize how to model single neurons, and I hope maybe next speakers focuses on more stochastic aspect of the neuronal processing. And let me start from the modeling a neuron. And to build a neuron model, we first simplify what is going on in the neuron. And this middle panel, this one, indicates what is going on in the neuronal membrane. And typically on the neuronal membrane, there is a ionic channel, which are special, selected, some selected, special, only selected ions can go through. And there is a repeat bilayer. So simplifying this part, we can consider simplified electric circuit model, which is sort of the parallel circuit. One consists of capacitance, which corresponds to repeat bilayer, and this resistance. So resistance corresponds to ion channel. And we can derive very simple linear first order differential equation model. And but it's too simple, and because this model is linear and does not generate any spikes, so we have to modify this model to generate to make a neuron generate spikes. And one more essential feature is the neuron has a spike threshold. It means if the neuron generates spikes, it means when the voltage reaches to some threshold here, the neuron generates spikes. So by combining this linear model and the threshold model, we can develop a very simple. It is a very famous model. It is known as a Leak integrated fire model. Essentially, this model consists of integration of input and the spike threshold. And by simulating this model, we can generate a sort of the we can simulate periodic output in response to a constant current, like this. But unfortunately, there are diversity. There are many types. There are various types of neurons, and so if this model is correct, all the neurons should fire regularly to this constant current. But nature is not so simple. So you can see this shows examples. So this is in this panel the experiment did injected various neurons, and they did experiment on typical 4 types of neurons: regular spiking, and fast spiking, and chattering, and bursting neurons. And all the case, the experiment that is injected constant current and no stochasticity there. But you can see from this figure, all the neurons, most neurons does not generate regular spikes, except for this fast spiking neurons. And regular spiking neurons, they initially generate frequent spikes, and but it goes to regular and tone down and regular spiking. And chattering neurons repeated sort of the bursting spikes. And intrinsic bursting spikes initially generate spikes with very high frequency. It is called bursting, and they going to silent. And it's unclear for how this neuron generate these kind of mechanisms. And today we will focus on 3 questions. And first, we want to think about how this neuron generate this variety of response patterns. And then we think about how this property is implemented in physiologically. And finally, we think about how this adaptation property is useful for neuronal coding. But today, mainly I will focus on these 2 questions. And let's start from the first one. And so to develop a simple model, and we think very fast, we start from a phenomenological model, and it is called a multi-time scale adaptive threshold model. And in this model, it's quite phenomenological, and we just extend the original, integrated fire neuron, the threshold is a constant. But this model, we extend this model to a variable, time-dependent variable. And the threshold increase after each spikes, self-generated spikes. After each spikes, the threshold of the neuron increased the sum of exponential functions. Typically, we consider 2 time constants, slow and fast, and increase and decrease exponentially. So this is very simple model. And we have 3 parameters as constant, and the weights of the fast and the slow, the coefficient of the fast and the slow component of exponential function. Okay. And but though it is very simple, but interestingly, this model can achieve a very high predict accuracy. So this is an example of the experimental data, and so the top panel shows fluctuating current, and this red one is a response to actual 3 neurons. And in this experiment, we did 3 types of neurons, regular spiking and bursting and fast spiking. And the red one is experimental data, and the blue one is prediction by this MAT model. And you can see from this figures, you can see MAT model can predict firing patterns of these variety of neurons. And if we examine carefully, quantitatively, we can see this model can achieve approximately 90% of spikes with error of 2 milliseconds. And furthermore, so there is an international competition for predicting neuronal responses as accuracy as possible. And our model won these competitions. And not only that, recently we explore various examine various experimental data, and interestingly, in olfactory neurons in insects, this adaptive threshold model is a very sort of the adaptive threshold mechanism is essential to reproduce experimental data. And for example, this left panel shows receptor neurons in mouse, and the receptor neurons is fast neurons. When we sniffing the smell, it is the fast neurons. And so there is a comparison, and you can see the dashed lines are experimental data, and this green and this real lines is fit by the model. And you can see the LIF model cannot fit this experimental data, but the adaptive threshold model, they can accurately reproduce these experimental data. And not only the first order neuron, interestingly, the second order neuron, projection neurons, it's a second order. So the first neuron is a receptor neuron, and the information is transferred to the projection neurons in the insect olfactory system. And we can do the similar analysis to the experimental data, and we can also confirm this adaptive threshold is essential. Spike frequency adaptation is essential to reproduce this experimental data. And but some question arises. So this model is so good. So can this MAT model reproduce everything? And unfortunately, it is not. And this MAT model cannot reproduce all the response patterns proposed by Izhikevich, and who developed a mathematical model based on bifurcation theory. And but if we extend this model, we can reproduce all the response patterns proposed by Izhikevich. And what is missing? The missing component is a voltage threshold, voltage dependent threshold. And we briefly explain the model, and the difference of the, so by incorporating this voltage dependent threshold, we can propose an augmented MAT model. And the key difference is this term. And this is the theta is again the threshold, but theta is dependent on the derivative of the threshold. And this is essential to reproduce remaining variety of response patterns. But if we incorporate this term with spike frequency adaptation, even though this model is sort of the piecewise linear system, but this model can reproduce all the response patterns proposed by Izhikevich. And then we go to the second question. So, but we focus on the first adaptation term in this talk. The second question is what is the physiology underlying this spike frequency adaptation? And to think this question, we consider the more biophysical neuronal model. It is a nonlinear differential equations and consists of sodium, potassium, and slow potassium, and the calcium dependent slow potassium current. And this is a spike generation current, and this is a slow potassium current. And we cannot talk about the details, but by approximating the spikes as pulse and using linearizing approximation, we can somehow derive reduced mat-like neuron model from this Hojikin-Huxley model. And interestingly, this term consists of 3 terms in the spike frequency adaptation. And this first term, it's a fast time constant, which corresponds to the fast sodium and the potassium. It corresponds to the original Hojikin-Huxley model. And this second term, it consists of M current. And interestingly, this M current, the time constant depends on the V voltage. And you can see the last term is AHP. And we can summarize this part by comparing to other models. And this slide, we compared the proposed MAT model with Hojikin-Huxley and the integrated amplifier model. First, we compare the prediction accuracy. And while these conventional models can predict 55 or 60%, the proposed model can predict around 90%. So it achieves a very high accuracy. And as we explained before, the MAT model can also reproduce diverse firing pattern. And the nice feature of the MAT model is that there are only 3 or 4 parameters. And in terms of speed, speed is computational speed. It is also important for the brain simulation. And we can show this proposed model is comparable to the LIF. So it's quite fast. And finally, we explained before, the MAT model is also, we can show it's connected to Hojikin-Huxley. So it's slightly worse to this model, but the MAT model is also interpretable to some extent. And we can also compare this with Izhikevich model. And unfortunately, Izhikevich model, it's difficult to quantitatively fit the model because the Izhikevich model is inspired from the bifurcation analysis. So bifurcation structure is a topological feature of the model. So it's very difficult to fit statistically or quantitatively. So that's a difference between 2 models. And another difference is computational speed. So because this Izhikevich model is a nonlinear model, so it's difficult to develop, simulate efficiently. And of course, interpretability is not, we don't know how to connect to Hojikin-Huxley model. And so we can conclude the MAT model is suitable for simulating brain. And finally, we don't have we don't have not so time, but finally we slightly explain how this spike frequency adaptation can be useful for neural coding. And it's sort of the ongoing work. And we are now sort of the modeling the projection neuron, second order neuron in the fruit flight. And we are doing simulation. And this is input firing rate to the neuron. And this is the output firing rate to the neuron with adaptation. And this one is without adaptation. And we consider second layer neurons, input and output, just a very simple neural network. And we can see that with adaptation, the response achieves sort of the peak. But without adaptation, so the response just copying the input. So this result suggests a spike frequency adaptation facilitate the neuron to detect the changes in the input. And also, we don't talk about in this presentation, but another feature is another feature of the sort of the spike frequency adaptation is that maybe it is also useful for saving energy, as Lubomir Koshtal showed, sort of so it improves the information transfer performance per spike or per energy. So this is another aspect how spike frequency adaptation is useful in the neural coding. And I also personally think maybe it's in future work, it will be very interesting to implement this sort of the spiking neural network with adaptation to developing sort of the neuromorphic engineering and to investigate further about the neural coding. And finally, I summarize. I talked about the 3 research questions. And the first question is, how does a neuron generate a variety of response? And this is a key feature is a dynamic spike threshold. And the second question is potassium currents. And finally, so the spike frequency adaptation can be useful for change point detection, but need more works. Thank you very much.
Thank you, Ryota. Questions? I see a question on the chat. What did we learn about the brain from your model?
That's a difficult question. But one aspect is spike frequency adaptation is implemented most of the excitatory neuron. And I think the reason is maybe the brain is very severe to save energy. So like not rich people, we are. So neurons are, so neurons are very serious to save energy. That's the reason maybe most of the neurons have a spike frequency adaptation.
Are there further questions or comments?
I do have a question.
Thank you. Yep.
So Ryota, you mentioned about the convenience of your model against the leaky integrated fire model. If I remember correctly, in your model, you have a term that is proportional to the first derivative of V, and that's a sort of memory term, right?
Augmented version.
And now at some point you had like a bit with an integral over when you
this one.
Isn't it? So if I were naively to implement that term, I would expect that to be really a time-consuming thing. So how did you get around that so as to make your performance as good as the Leaf model?
Yeah. So, but there, we assume. So I'm not this. I'm not the author of this paper, but yeah. So this the key point is we assume special kernel function for K. Of course, you are right if we assume general K, but we assume exponential function or something like that this integral can be semi-analytically solved. So this equation can be transformed to a system of linear differential equations.
So you have effectively an additional linear equation as opposed to
Yes,
compute.
linear equation for theta.
Thank you.
And the linear equation can be analytically solvable. That's a key point, yes.
I see another question from William Brown. In the last part, you showed how a neuron with SPA can detect the input change in its firing threshold. How does this depend on the adaptation timescale? And can you use that information to figure out which current likely cause SPA in that neuron?
It's a good question, but so sorry, we didn't have investigated detailedly about the dependency on the time scale, but yes, I think this so this is in general this property is robust to the small changes in the time scale, but of course, if it is too large or too small, maybe the performance can will drop.
Yeah. So
Yeah.
I mean, you see that there is some sort of decay, right? From this peak, there is a decay, like
Yes.
in this, and then I mean, there exactly, yeah. So relaxation to some constant firing rate. And I mean, I would say
Yes,
that probably depends on the adaptation MAT.
Yes, you are right. You are right.
And
yes,
and because previous, so basically I asked this question because previously you showed that different currents give different time scales, and I just thought that okay, maybe this is a way to say which in this model is or which in this neuron, because it's a biophysical thing, right? Is that work?
Yes. Thank you very much. And this in this case, this time constant is fitted from a fruit fly neuron, and the time constant is quite relatively I guess the time constant is approximately comparable to the calcium.
Okay.
Yeah.
Thanks.
Thank you very much.
I think it's time to stop and to move to the next speaker.
Yes, I will.
I'm sorry for the other questions that were not answered.
I will reply on chat.
Okay, perfect.
Yeah, thank you very much.
Thank you, Ryota.
Thank you very much.
Our next speaker is Rimzin Tomar from Academy of Science in Prague. And she will speak about increasing interspike interval variability can yield a decrease in instantaneous firing rate dispersion. Please, Rimzin.
Hello, thank you so much for the introduction. You can see my screen, yes?
Yes, we do.
Okay, wonderful. So the title of my talk is Increasing Interspike Interval Variability Can Decrease Instantaneous Firing Rate Dispersion. This work was done with my supervisor, Professor Lubomir Kostal, and I will, So I'll start with the outline. First, I'll talk about spike trains as stochastic processes. Then I'm going to talk about the statistical analysis of stochastic neural activity. And here I'll introduce the concept of instantaneous rate. And then I'm going to talk about the statistical dispersion measures that we're going to use for our analysis. After that, I'm going to talk about the results where we applied our proposed methods to the standard ISI models. And we considered 4 standard ISI models, as well as we also applied our results to some experimental data. After that, I'll talk about the conclusions of what we found out. So to start with, I don't know how it works. Okay. So to start with, I'll first talk about the spike trains as stochastic processes. Right, so the class of stochastic processes whose realizations consist of a sequence of point events in time is called stochastic point processes. And spike trains, they are described as stochastic point processes where the spikes, they correspond to the events. So the spikes or the action potentials, and those are the events. So for any stochastic point process, there are two ways of describing it. We can describe it in the terms of number of events occurring during a time window, how many events occurred in a particular time window, or in terms of the interval between these occurring events. So consequently, to describe a spike train, we can also use these two different ways of description. Either we can use the sequence of spike times, which I have termed here as x1 and x2, or we can describe the spike train through the interspike intervals, which we define as the time that is between two consequent spikes. So here is a graphical description of a spike train. The interspike interval is termed t, and it belongs to the probability distribution f of t. And it is the time that is between two consequent spike, xi and xi plus 1. Going further, renewal processes. That's what I'm going to describe on this slide. But before that, I'm going to talk about a class of stochastic point processes called stationary point process. So a stationary point process is the point process where the joint distribution of the number of spikes in any k time intervals, it doesn't depend on the displacement variable h that you choose. So it doesn't matter which h you choose, the joint probability distribution is going to be the same. Now, an important subclass of this stationary point process is called the renewal point processes, in which the length t of consequent interspike intervals is an independently and identically distributed random variable. And the probability distribution I've termed here as f of t. Now, in the talk, I'm going to talk exclusively about renewal point processes, and they're often used to model the activity of spontaneously active cells. Now, in renewal point processes, there are two very important subclasses of it. The first subclass is called equilibrium renewal process. So, equilibrium renewal process, in this, the time origin where the process starts, it doesn't coincide with an event. So say xi here is the spiking event, but the origin starts at a random time that is unrelated to the spike times. This is termed the equilibrium renewal process, as opposed to the ordinary renewal process, in which the origin time, it coincides with the spiking event. So these are the two important subclasses of renewal processes. We're going to focus on equilibrium renewal process, and why I'm going to get to that in a minute. But first, let me jump to the next section, which is the statistical analysis of the stochastic neural activity. Right, so let me introduce the concept of the instantaneous firing rate. It is typically defined as the inverse of the interspike interval t. However, as Lansky and colleagues showed in their 2004 paper, the mean of the instantaneous firing rate is higher than or equal to the mean firing rate, which is also equal this 1 over E of t. This is the mean firing rate, and it also equals to the point process intensity. So this is the inequality that persists, and that's why you can't use the mean of the instantaneous firing rate in place of the mean firing rate generally. However, this inequality, it disappears if you remember that the instance where you want to observe the instantaneous firing rate, it generally does not coincide with a spiking event. I'll explain that further. This is a graph that's taken from the paper that was published by Kostel and colleagues in Chaos Journal in 2018. So what do I mean when I say that the instance where you observe the instantaneous firing rate doesn't coincide with a spike? All right, so remember we talked about the equilibrium renewal processes where the time origin does not coincide with a spiking event. The same principle here, we have different spike trains here, and the instance where we observe the instantaneous rate given here, the observation time titled T0, it's unrelated to the spikes. It's not in sync with any of these spike trains. So in this case, the interspike interval that we observe here, they are going to, the probability of us observing an interspike interval is going to be proportional to the length of the interval. In simple terms, the longer the interspike interval is, the higher is the probability that we're going to encounter the interval. Right, in this case, the interval that we observed, these are termed the length biased intervals. Yeah, because their length actually impacts if we're going to observe them or not. So these are depicted as T tilde, and in this case, the instantaneous rate that we have, I'm going to call it R. So R is equal to 1 over the length bias interval instead of, just being 1 over interspike interval. So in this case, the R that we have, that's going to satisfy this equality, which means the mean instantaneous firing rate, E of R, is going to be equal to the point process intensity. So this inequality that we had, in case of observing the length bias intervals, it turns into an equality. So for the rest of the presentation, when I talk about the instantaneous rate, I'm talking about R, which is the inverse of the length bias intervals. So keeping that in mind, let me talk about the statistics that we're going to look at in this talk. So I'm going to talk about interspike interval statistic, and I'm going to talk about the instantaneous rate statistic. So interspike interval is going to give us the temporal perspective of the spiking data, and the instantaneous rate statistics is going to tell us about the rate coding perspective. But in a way, the instantaneous rate statistic is going to tie into the rate coding perspective because it's instantaneous rate, and also the temporal perspective, because by definition, this takes into account the spike timing. Right, so we're going to talk about the variability and randomness of these two quantities. And for variability, we're going to use the coefficient of variation. And for randomness, we're going to use a CH, which I'm going to expand on further. But first, I want to talk about the variability and randomness. So variability and randomness, these are two very similar quantities that we use to further differentiate between the spike trains that might seem similar from the rate coding perspective. But it's important to distinguish between these two quantities, because we might have really variable data, but it might not be random at all. If, for example, if we talk about data from uniform distribution, right? So highly variable data might not be random at all. To expand on that further, let's look at these three instances. This is going to make it clear. So in the first instance, we look at that the data is really concentrated around the mean. So we have really low variability in the data. All the values are concentrated with negligible spread around the mean. So the randomness is low, as well as the variability is low. The randomness is low because we can easily predict what are the values going to be. In the second instance, we see that the variability is high, the peaks are far apart, but the randomness is low because the data is going to have really predictable values, either this or this. In the third instance, we see that the randomness is high, and because the spread of the data is wider, the variability is high as well. Right. So after this, so we have the variability and the randomness. After this, I'm going to talk about the dispersion measures that we have used to quantify the variability. So the standard dispersion measure for variability that's generally used is the standard deviation of the ISIs. But here we use the relative dispersion quantity, that is the coefficient of variation of the interspike interval, which is defined as the standard deviation divided by the mean of the ISIs. The coefficient of, I'll just expand on this further. The coefficient of variation, we use this instead of the standard deviation because CV, it's a dimensionless quantity. So in case we have, it doesn't depend on the units of the interspike intervals, and it doesn't depend also on the linear scaling. And building upon this, we derive the definition for the coefficient of variation of the instantaneous rate. The derivation is given in the 2021 paper. So advantage of using CV is that for exponential distribution, CV of ISI is going to be 1, regardless of the mean of the distribution. Conversely, if you have CV with the value 1, that does not really imply the exponentiality of the distribution. So standard deviation or CV, it measures how much the PDF, how much the probability distribution is spread with respect to the mean value of the data. But however, whether it be standard deviation or CV, it doesn't really quantify how random the values are. So even with even with the standard deviation and CV, we still don't cover all the ways the spike trains can be different from each other, even though they might seem similar from the rate coding perspective. And that's where the randomness comes in. So the dispersion measure that we use for the randomness, to quantify the randomness, as a further describing characteristic of spike trains, we use the entropy-based measure, and that's called the differential entropy, which is defined here for a continuous random variable x. But we don't use h by itself as the dispersion measure here, because h can take either positive or negative values, and also it also depends on the scaling. So we really need a relative measure. So to get there, we first consider the entropy-based dispersion coefficient of randomness called the sigma h, which was derived in a 2013 paper by Kostel and colleagues, and it can also be seen in a 2010 paper by Kostel and Marsalek. Now we have the entropy-based dispersion coefficient of randomness. Using this, we can derive the relative entropy-based dispersion coefficient, which again is, we termed it CH, and it's achieved by dividing the sigma of h by the mean when it comes to ISI distributions. Similarly, the sigma h and CH for the instantaneous rate are, they're defined analogously. We just change T to R here, and the definitions remain the same. So unlike the coefficient of variation, when we have the value of CH as 1, it only occurs in the case of an exponential distribution, and this is the maximum value of CH. So now we have, I have described what instantaneous rate is, and I have talked about randomness and variability, and I have talked about different measures that we are going to use to quantify the randomness and variability for the rate coding perspective and the temporal coding perspective, which means the T and the R that we talked about before. So now let's look at some results. So for results, we're going to consider the standard interspike interval models of neuronal activity. And we're going to restrict ourselves to the framework of renewal spiking activity. So for that, the first three models that we consider, that is the gamma distribution, log normal distribution, and the inverse Gaussian distribution. Right, so these are scale family distributions. The probability distributions are parameterized by the intensity, lambda, and they satisfy this peculiar property here. The fourth distribution that we will talk about is the distribution involving a refractory period. So first, I'm going to show you the results for the variability and randomness for these four distributions together. After that, we are going to take a look at something more complicated, slightly more complicated. That's the mixture of two distributions with that time. Analytically, this gets more complicated. But after that, we're going to look at what all of this actually means when it comes to experimental data and where we can apply the results, the analysis that we have seen in this talk. So let's start. First, let's look at the dispersion measures of variability. So we're going to first look at the first four models that we talked about. That was the gamma distribution, the log normal distribution, inverse Gaussian distribution, and the shifted exponential distribution. And we are looking at the dispersion measures of variability, which is the coefficient of variation of the interspike interval here on the x-axis, and the coefficient of variation of the instantaneous rate. That's here on the y-axis. Right, so let's start with the gamma distribution. So for the gamma distribution, as you see here, the coefficient of variation of the instantaneous rate, it is higher than the coefficient of variation of the interspike interval. And as the value of the CV of ISI, as it approaches 1, we see that the coefficient of variation of R, it approaches infinity. And it happens because as CV of T for gamma, as the value is 1, the gamma distribution becomes exponential distribution. And consequently, the value of CV of R is infinity. And then if we look at the log normal and inverse Gaussian distribution, that's here, both of them, we see that the relationship between the CV of t and CV of r is that of identity. So nothing really changes if you jump from the ISI data to the instantaneous rate data. They both have similar dispersion measures of variability. And why does that happen? That's because for these two distributions, the log normal and inverse Gaussian distribution, the probability distribution functions of the instantaneous rate and the ISI, they hold this symmetrical property. So they have the same profile. If lambda is equal to 1, if we have the intensity equal to 1, the profiles of the instantaneous rate and interspike interval are going to have the same shape. So that translates to their CVs having the same profile as well. Then coming back to the shifted exponential distribution, where the relationship is not identity, we see that the coefficient of the coefficient of variability, CVR, it's less than CV of t until a certain value. And after that, the CV of t approaches 1. Right, okay. Now, so this was the relationship between the coefficient, between the dispersion measures of variability. Now, if we look at the dispersion measure of randomness for these four models, we see that the relationship is a little bit more complicated. So first, I'm going to talk about these three distributions. They are the scale family distribution. In the direction of the arrows, we have coefficient of variation of the interspike interval going from 0 to infinity. So if we start for the gamma distribution, this starts at 0 and it increases first and then it decreases. And it is the case for all these three distributions. So let's start at the beginning. So in the beginning, we have the coefficient of variation of the ISI is 0. So in that case, it's approaching 0, CHR is approaching 0 as well, and as well as CHT for these two distributions here. And as the value of CVT increases, the value of CHT and CHR both increase. The randomness keeps increasing until a certain point. After that, it starts decreasing. And as we have the CVT approaching infinity, both CHR and CHT for these three distributions, it approaches 0. Then we have the shifted exponential distribution. So for shifted exponential distribution, CHT here, it increases as a function of CVT. So as variability increases in the interspike intervals, the randomness increases as well. But if we look at the instantaneous rate, it increases the function of ISI variability up to a certain point, and then it starts declining. So this was the dispersion measures of randomness. The relationships between different measures are not that straightforward as you would expect. Now going back to one of the other models, the
mixture. 5 minutes included the questions, so 2 minutes in order to have questions.
Oh, okay. I'm gonna be really fast. So this was this was the mixed distribution and the theoretical part of it. All right. So with the mixture of two exponential distribution with refractory period signified as tau, and the mixture components are A and B. Theoretically, it was really hard to obtain the formulas for CV and CH, so we just simulated it with some parameters. We kept one parameter fixed and we changed the other parameter. And the relationship was really complicated, but this becomes clearer when we looked at the experimental data. So we looked at some experimental data that was obtained from the spontaneously active afferent neurons in the zebrafish lateral line. And I'm going to skip on the explanation of the spiking data, just that the ISI data found in these experiments, it was best fitted to a mixture of exponential distribution with that time, the one that we just looked at. So in this case, if we look at the CV of T and CV of R for this data, we see that the neurons, which might have similar CV of T values, which might have they can still be differentiated on the basis of their CV of R values. And we get similar results from the CH values. If you see here, these neurons if you look at the randomness of their interspike intervals, these neurons have similar values, but you can still differentiate among them on the basis of their CHR values. This confirms our assertion that the instantaneous rate, it provides us with a different or novel outlook on the spiking data that's different from instantaneous rate, and which is what I talk about in the conclusions, which I'm going to just share here with you, that there are different relationships between the variability and randomness measures. And for a given model of ISI, an increase in the dispersion measures of ISI may result either in an increase or decrease, depending on the model. So the statistics can provide us with a different outlook on neural data, and it's worth looking into. And thank you so much for listening to me. I'll end my talk with this. And if you want more information, you can look at our paper, which was published last year in Frontiers. Thank you so much.
Thank you, Imogen. We have time just for one question, if any. Otherwise, you can write in the chat and she will answer in the chat.
So I have one question, if it's possible. So I enjoyed very much, very nice presentation of this stochastic framework. What puzzles me is the following: you don't have a single point process, you have a system of interacting point process. Could you please say something about the way you present the system itself? Because, well, you have problems of existence and so on, the properties. Did you think about the description of the system of interacting point process like the one you presented?
It's actually not a system. I probably didn't present it as better as I could have. It's just different processes. I consider them independently. I just presenting them together so you can see how different they are from each other, the relationship.
Okay, thank you.
Thank you.
If there are any more questions, I would be happy to chat about it.
Thank you again.
Thank you so much.
And we move to the next speaker. She's Ola Shur from Kyiv University and from Budapest Institute of Experimental Medicine. And she will talk about the distribution of interspike intervals of a neuron with delayed feedback inhibition simulated with a renewal point process. Please, Ola.
Okay, thank you very much for the introduction. I am currently a PhD student at the Bogoliubov Institute for...
Can you increase the audio? We don't hear well.
Okay, 1 second, I will. Could you hear better now?
Yes, it is. It's better.
Okay,
Thank
great.
You. Please start.
Okay, thank you. So I'm a PhD student at the Bogoliubov Institute for Theoretical Physics in Kyiv, Ukraine, but as I have been said, I currently at the Institute of Experimental Medicine in Budapest in Hungary. And today I would like to present our joint research with my PhD supervisor, Alexandra Vidybida. So we will be talking today about the impact of delayed feedback inhibition on neural activity. And why is this topic important? So as you may know, for brain, in order to function properly, there must be a balance between excitation and inhibition. However, a lot of recent experimental evidence indicates that cortical disinhibition is important for many cognitive processes like learning, memory, attention, social behavior, and so on. And cortical disinhibition is a transient ceasing of inhibition or activity of inhibitory neurons. And this cortical disinhibition can be achieved in different ways, for example, at the level of local circuits. And in one of the cases, how this can be achieved at the level of focus circuits. For example, one type of inhibitory neurons, that is somatostatin expressing interneurons, they are inhibited fed forwardly by other type of inhibitory neurons, parvalbumin expressing interneurons. However, it is unclear how the inhibition of parvalbumin expressing interneurons is achieved. However, recent work by Deleuze and colleagues provides us with a possible answer to this question. And the previous experimental work showed that parvalbumin expressing interneurons in the cortical areas tend to send synapses not only to other neurons, but also onto themselves. And these self-synapses are called autopsies. So Deleuze and the colleagues showed that for parvalbumin interneurons, the main source of inhibition is autoptic self-inhibition. And all of this experimental evidence motivates us to study the impact of inhibitory autopsies, or in other words, delayed feedback inhibition on neural activity. And we approach this research as follows. So we consider some class of non-adaptive spiking neuronal models. And we do not specify the exact neural model. However, we specify the conditions of the set of neural models that we consider. And I will talk about this set of neural models in more detail a little bit later. So we consider that some neuron is stimulated with a random sequence of identical excitatory impulses, and this sequence is random, and under this stimulation this neuron generates another sequence of spikes. And also, since we are interested in neurons with inhibitory autopsies, we assume that every time neuron generates a spike also enters the delayed feedback line or autops. And the impulse in the feedback line needs delta units time to reach the neural input and act somehow on this neuron. So as regards the input stimulation, so as I said, we consider that this is a stream of identical excitatory impulses, and we assume that this stream is a realization of some stochastic renewal point process. And we assume that we know the distribution of interspike intervals for this input stream, and we denote this distribution as P(in) of t, where t is an interspike interval duration. And our goal is to derive the distribution of interspike interval for the output stream for this neuron with delayed feedback. We denote this distribution as P of t. However, we would like to express this distribution P of t in terms of the distribution for that same neuron under this same renewal stimulation, but without delayed feedback. And this distribution for such neuron we denote as P0 of t. So to sum up, we assume that we have distribution for input stream P(in) of t, that we have distribution for a neuron without delayed feedback P0 of t, and also that we know the delay of the feedback delta, and we are looking for the distribution for a neuron with delayed feedback P of t. So as I have promised, now a little bit about the set of neural models that we consider. So this set of neural models include such popular models as perfect integrator, leaky integrator, fire neuron, and also binary neural model. And we assume that the class of neural models that we consider is it satisfies these 5 conditions, which includes that neuron is deterministic, that it can be triggered only at the moment of receiving an input impulse, and also that just after triggering, neuron fires a spike and immediately it appears in its resting state and remains there until an input impulse is received. Also, we assume that we know the distribution for renewal input stream by n of t, and also the distribution for output stream p0 of t. As regards the delayed feedback inhibition, we assume it has these 3 following properties. The first one is that the time delay in the feedback line delta is constant. The second one is that the feedback line is able to convey no more than 1 impulse at a time. And the third one is that after receiving an impulse from the feedback line, the neuron appears in its resting state and that impulse is immediately forgotten. So now let's move to how we approach the finding of a distribution of ASIs for a neuron with delayed feedback. But firstly, I want to emphasize 2 things. That the first one is that we study neural activity in stationary regime. And the second one is that under all the assumptions that we have made, always at the beginning of the interspike intervals, there is always an impulse in the feedback line. And let's say the impulse in the feedback line needs the beginning of the interspike interval. This impulse needs S units of time to reach the neuronal input. And we will call this variable S as time to live. So now we can find the distribution of ASI for neuron with delayed feedback using this expression. And here there is integration over different times to live. F of S is a probability density function of times to live as in the stationary regime. And the second distribution is a conditional probability density to get the output in ASI of duration t, given that at the beginning of the ASI, there was an impulse in the feedback line with time to live s. So now let's find the expression for these 2 distributions. So firstly, let's talk about the distribution of times to live f of s. So we have proved that if this condition on the distribution for a neuron without feedback, P0, and the delay of the feedback delta is met, then for any initial conditions, stochastic activity approaches a stationary regime characterized with such distribution of times to live f of s, which consists of regular part and singular part. And here you can see the expressions which will help to find these different parts of the distribution f of s in the stationary regime. So as regards the conditional probability, with this conditional probability, the expression for it you can see here. Here, chi is a Heaviside step function. Here is a distribution for a neuron without feedback. Also, here you see one of the 2 auxiliary distributions that we need to find our distribution. So this one is a distribution of output ASI for a neuron with excitatory instantaneous feedback. And this distribution you can find solving these integral equations. And the second auxiliary distribution is this p tilde 0, and it allows to calculate the probability of obtaining an input impulse at time s prime that will not trigger the neuron without feedback. And this auxiliary distribution you can find solving this integral equation which requires us to know the distribution of input ASIs and also output ASIs for a neuron without feedback. And one thing I want to note here is that if we consider as an input Poisson point process instead of general renewal point process, this expression simplifies a lot. And you can see here how it looks in case of when its input process is Poissonian. And the reason why is that is because if you look at the intensity of point process, when the point process is homogeneous Poisson process, intensity is constant. However, it's not the case for the renewal point process, because intensity of renewal point process depends on how much time passed after the last event. So returning to the distribution of ASI for neuron with delayed feedback inhibition, this expression for this distribution, P of t, is different for different values of interspike intervals. So we have this expression when ISI duration is less than delta, and we have this expression when ISI is longer than delta. As we can see, the resulting distribution has a jump type discontinuity at the point that corresponds to ASIs equal to delay as a feedback line. And to illustrate these general analytical formulas, I would like to show you the results that we have obtained for the model of body neuron with threshold 2 under Erlang 2 input stream stimulation. So here in the upper left corner, you see the plot of the distribution for input stream. So interspike intervals here are distributed according to Erlang 2 distribution, and you can see the plot here. Here on the right, you can see the distribution for the neuron without any feedback. And here on the left, you see the distribution for a neuron with delayed feedback inhibition. And in this case, we set the delay of the feedback at 2.5 milliseconds, so at this point, and you see there is a jump type discontinuity at the distribution. And this jump type discontinuity makes it impossible to model this distribution as any simple distribution. And also here you can see the plot of the results that we obtained by numerical simulation of stochastic neuronal activity to verify our analytical results. So to sum up, like two important points of my talk are that the first one, that you can find the distribution of ASIs for neuron with delayed feedback inhibition if you know the distribution for input renewal process, distribution for output ASIs for the neuron without any feedback, and also if you know the delay of the feedback, and the resulting distribution for a neuron with delay feedback will have a jump time discontinuity. So if you are interested, you can look at our preprint at Archive to find out more details of this work. So thank you very much, and it will be my pleasure to answer any of your questions.
Thank you, Ola. Are there questions or comments? We have time for answering.
There is a question from Cindy.
Okay, please, Cindy.
Sorry, can you repeat, please, one more? Because my connection
I like
is not
your talk very much. Thank you, Olha. I like your talk very much. And I was wondering about the delay being just through one neuron. It occurred to me that it would be interesting to find out what happens if the delay is actually through a sequence of neurons as well.
Thank you very much for your question. So depending on which sequence of neurons you consider, for example, you can consider, so in our work here, we considered an inhibitory neuron with self-feedback, but instead you can consider, let's say, excitatory neurons that is connected to some other inhibitory neurons that is connected to these original excitatory neurons. And I think under some conditions you can use our results to describe the activity of these excitatory neurons that is connected with inhibitory neurons.
Thank you.
Other questions, comments? I can ask something. Have you tried to check on data your model?
Mm-hmm. Yes, thank you very much for your question. So we have not tried to check it on the experimental data because, so the thing is, if you look at the experimental data that that studied the of inhibitory neurons, you can see that this delay is very short. It's usually less than 2-3 milliseconds. And if we look at the resulting distribution, we see that the peculiarity that is
we don't hear anymore.
Means that for these short interspike intervals, you can have like other factors that will influence this distribution. So that's why we have not tried to apply this result to the experimental data yet. And also, there is not a lot of experimental data about autopsies yet.
Thank you. Other questions? No. And we thank Ola again. And we have 3 minutes of advantage. Maybe we can start to share the screen, Giuseppe, but
start sharing and maybe
yes.
I wait.
But I suggest to wait the exact scheduling to start because maybe someone move from one tunnel to the other.
Sure.
So since we have 3-4 minutes, if anyone has comments or questions for previous talks, we can use this few minutes for it. I see something in the chat. Nothing. So we simply wait the exact scheduling. Rimijin is asking if anyone will join fence.
Just me.
Yes, please.
It seems so. Just you.
No one.
It would be nice to see people in person sometimes.
Yeah. Hi, Cindy. Nice to see you now.
We cannot hear you.
Oh, I said greetings to everyone. I see several friends and I'm very happy to see all of you. It's great.
The same holds for us, Cindy. And we appreciate your presence on, I don't know what time is in Canada, but I think it's not easy for you to participate.
Actually, I was with you already at 5 a.m. here.
I understood. Okay, I think now is time to start. Our last speaker is Giuseppe D'Onofrio, who from Torino University, and he will present the Jacobi process with jumps as neuronal models. Please, Giuseppe.
Okay, first of all, I would like to thank the organizers and Laura for organizing this session. It's nice to see you all. And today I want to talk about diffusion processes with jumps, in particular Jacobi process, and now these processes can be useful in the neuronal modeling. This is a joint work with Pierre Paty and Laura Sacerdote. Okay, so this is the kind of conference where I don't have to explain what a neuron is and how it works. So I also have very nice introduction by Ryota. Thank you for his introduction on the leaky integrated and fire model. So for me, it's much easier now. So I just have to explain how I intend to model the activity of a single neuron. Okay, for me, the voltage will be a stochastic process. And this is the time evolution of my stochastic process. And I'm looking for the distribution of the spikes. Okay, this is my main goal. And from the mathematical point of view, how the model works. The voltage is a stochastic process that evolves in the presence of a threshold. Consider here constant threshold. And the neuron receives excitatory and inhibitory input. And as soon as the stochastic process touches this threshold, we have an action potential. So we have one of these spikes. So the dynamic in this model, we assume that after the spike, the stochastic process is reset instantaneously to the starting position and the dynamics start anew. So I don't have refractory period here, and all the interspike intervals, so the time between the spikes, are independent and identically distributed, and they all distribute as this random variable t, so the first passage time. So I will focus on the first passage time problem for this kind of processes. Okay, I defined roughly how the model works. I said I want to use a stochastic process, but which stochastic process should I use? Okay, I will start with the classical case, no, the leaky integrated fire in its easiest form. So we have that the voltage of the membrane potential, so the membrane potential evolves in time according to this differential equation. So we have that tau here is the membrane time constant, and at some random times, described by Poisson process, n of E and of I, there is an excitatory or inhibitory input that will increase or decrease the voltage of an amount delta. Okay, so in this framework, you can assume that the jump amplitudes, so these inputs, so this delta are small enough, and the frequency of these inputs is very high, and we can take a diffusion limit. Okay, long story, very short. And the diffusion equation we obtain is an SDE of this kind, and the process describing the voltage now is this process Y, that is an Ornstein-Uhlenbeck process. This is very nice process because it's for its mathematical tractability. It has been used for decades. The literature on this process is huge. There are results on the first passage times, but this process has many drawbacks, my point of view. So there are many missing features in this model. So first of all, the Onsa-Lohner-Lembeck process has unlimited state space. It means that we are describing some physical quantity with some quantity that can take infinitely large values. That this is not possible. Moreover, the changes in the voltage should depend on the actual state of the voltage, and this is not taken into account by the Onsa-Lohner-Lembeck process. Moreover, we don't take into account the geometry of the neuron, and we are assuming that all the inputs are small and the frequency is very large. Okay, we want to overcome some of these features, and for instance, we can start from the first two. That's why the Jacobi process is used. Was proposed by Lansky and Smith, and then by Massimiliano Tamborrino, Pat Lansky, and I. So how this process looks like? It's a diffusion process again, and here I wrote it through is infinitesimal generator. The reason why I use this notation will be clear later. It's just that this is a Markov process, so I can define it using his infinitesimal generator. What is important to understand is that this part will account from the drift part of the equation, okay? And this is the part of the diffusion. So we see now that the diffusion part, that's the part describing the noise, the noisy part, depends on the state of the process. We see X. Moreover, we still have two processes that describe the arrival of excitatory and inhibitory inputs. But now we consider that the increment or the decrement of the voltage will depend on the actual state of the process. Okay, so this is the second feature we were looking for. And moreover, we observe that the state space of the Jacobi process is limited. Okay, the state space is VI, VE, where VI and VE are the reversal potential. So the inhibitory reversal potential and the excitatory reversal potential. Okay, so the first two features we were looking for are satisfied. And of course, we prefer to rewrite everything. We want to rescale everything in 0, 1, just to simplify the notation. Okay, so this is the Jacobi process that I will call the classical Jacobi process throughout the talk. I want to stress that I will devote half of my presentation just on the motivation. And then in the end, I will show the results. Okay, so we have this process and that we investigated these two papers. But going back to the first slide, my question was to analyze the first passage time problem for this process. Okay, so the best would be have the density or the distribution of T. We don't have it, unfortunately, in the case of the Jacobi process, or if you want the Wright-Fisher process, it's often also called Wright-Fisher, it's not known in a closed form. Okay, there is no analytical expression for the first passage time of the Jacobi process. So if you don't have the density, what you can try to do is to find the Laplace transform of the density. Why? Because if you have the Laplace transform, first of all, you can try to invert it. And unfortunately, you cannot do it in this case analytically. So no chance to invert this expression. But if you have the expression, you can find information on the tail of the distribution, or you can derive this expression and try to find information on the moments of the density of this first passage time. Okay, so for the Jacobi process, what I call classical Jacobi process, the Laplace transform of the first passage time is known to be a ratio of two Gaussian hypergeometric functions. Please remember these two functions because they will come back later. Okay, so it's a ratio of two hypergeometric functions. We cannot invert it, but we can derive it. Okay, we can derive it. We obtained, for instance, the first three moments. In particular, here I wrote the first moment that we are interested in because I'm using, according also what Rim Jim said before, we are interested in the instantaneous firing rate. And here we are using the definition that is the reciprocal of the first moment of the first passage time. Okay, so the instantaneous firing rate is 1 over this quantity. Okay, so this is a quantity of interest. We also found information on the variance. We evaluated and we found coherence, resonance, some the contribution of inhibitory inputs, many nice results on this process we have obtained. But if we go back to the first slide, we see that something is still missing. And I want to add something more. Imagine you want to account for external factors, like, I don't know, the neuron under study, the individual under study is under pharmacological treatment, or it's under drug or alcohol consumption. So you want to account for large external factors, for instance. How can we introduce also these features in our model? That's the reason why we propose to use a Jacobi process with jumps. Okay, so these jumps are different from the other jumps. These are more large jumps and are state-dependent jumps because we want to keep the second feature again, also with the jumps. So what is the process we are thinking of? The process we are thinking of is this one. Again, I'm writing it using the infinitesimal generator. We proved that it's a Markov process, so we can do it. And unfortunately, it's the only way, it's the best way to show it. Because if I try to write an SDE whose solution is this one, it's very unpleasant to read. Okay, it's not intuitive at all. I prefer to use the infinitesimal generator. So this new process that we call non-local process or Jacobi process with jumps, the generator is like this. So you have a term that is exactly the generator of the classical Jacobi, and we have jumps. Okay, the jumps are according to a measure pi. What is important to understand now is qualitative behavior of this process. So first of all, I want to stress that the jumps are state dependent, both in the frequency, because x is here, and in the size here. So what happens? That if x, so here the process is rescaled in 0, 1. Okay, so if x approaches 0, so the lower boundary, then the frequency of the jumps increases. So we have a lot of jumps, but the amplitude of the jumps becomes smaller and smaller. So we have a lot of small jumps close to the lower boundary. If we are close to the upper boundary, well, we have less jumps, but the size of the jumps depends on the measure pi. Okay, so if pi is a measure that has a lot of mass around 0, then we will have mostly small jumps. If pi is a measure that has mass also for high values, we will have also large jumps. In any way, the jumps here are only downward, and then I will explain why. And even if the jump is large, it will never go below 0, okay? Never go below the inhibitory reversal potential. Why downward jumps? Because we had in mind this application to the pharmacological treatment, or we will see at the end that also maybe adaptation can be treated in this way. So imagine now that we have downward jumps and are state dependent. And with this plot, we should understand even better how the dynamic works. So here we have possible trajectory of this Jacobi process with jumps, and here we adjust the count of the jumps. Okay, so here we have the first jump, and we see that there is a downward jump here that is pretty large. Here another jump, another downward jump. Here in this plot, the lower boundary is -10, you see here, and the upper boundary is 100. So we see that if we go closer to the lower boundary, the number of jumps is increasing. So we have a lot of jumps, but the size of the jumps is smaller. Okay, so this is the behavior of our process. To ensure that all the dynamic will stay above the lower boundary. So it means that inhibitory reversal potential Vi is an entrance boundary. We have to post this. It means that the drift compensate in some sense the downward jumps and the noise. Okay, so this is our very important assumption. Okay, so now before going to the main result, I need to introduce some ingredients, mathematical ingredients, and then we'll see the main result. This kind of process I defined here, this one, it's a Levy type process. And all the Levy processes are defined by a triplet. So here it's this lambda. In lambda, there is the time constant, membrane time constant. Mu accounts from the input, and pi is the distribution of the jumps. Okay, so the process and his generator is described by this triplet. And there is a one-to-one correspondence between this triplet, and a Bernstein function. What is a Bernstein function? It's just a function that is a positive function, infinitely differentiable with this property. Okay, what is important to say is that if we have a Levy process, we can write a unique function, Bernstein function of this kind. Okay, so in our case, this Bernstein function will have this form. And it depends, of course, on the distribution of the jumps, pi. Okay, why these Bernstein functions are important, we will understand it later. But first, I want to stress what can we define with this function phi. This function w, here is evaluated when n is an integer number. So it's the product of this function phi. But in general, if n is not integer, we can define it as the solution of this equation. This is a function that was introduced by Paty and Savov in 2018, and it's a generalization of the gamma function. You see that from this definition that if phi n is equal to n, actually this is the definition of the gamma function. So this extends the gamma function. And using this extension of gamma function, we want to extend our Gaussian hypergeometric function. Do you remember that the Laplace transform, or the first passage time of the Jacobi process, was a ratio of two Gaussian hypergeometric functions of this kind? And that is actually, it has this expression without the w. If we add this w here, of course, it's a new function, but we call it still using this notation to phi1, phi, because if we don't have jumps, so if pi is equal to 0, we don't have jumps, actually the two functions coincide. Okay, so it's a way to generalize the Gaussian hypergeometric functions. Okay, now we have the list of all the ingredients, and we are ready to read the main result. We were looking for the first passage time of this process, and we were able to write the Laplace transform of the first passage time of the Jacobi process with jumps. And look, it's again a ratio of two functions. It's actually a ratio of two generalized Gaussian hypergeometric functions, where the function is the one that we defined here using the Paty-Savov function. Okay, this result is nice, and I just want to stress that it's not trivial. It's true that we obtained something similar to the classical case, but in some sense, we force it to look similar. No? So it's not trivial at all. I will not go into the details of the proof because it's very technical and very long. I will just want to show one step of the proof because it gives you the flavor of what's the idea behind this strategy. So the idea is to inherit all the information we know on the classical Jacobi process to the case with jumps. Okay, so we built an operator, this lambda, that will map our information that we know on the classical Jacobi process to the case of the Jacobi process with jumps. Okay, how can we build such an operator? We use a theory of Levy processes and subordinators, because in particular, lambda is defined this way. We know, I said that to a Levy process, we have a Bernstein function, a correspondence to a Bernstein function. To a Bernstein function, we have a correspondence to a subordinator. That is a stochastic process that is non-decreasing and with stationary increments. What is important so is that if we know the Bernstein function and we know it, we are able to write it, we can obtain the subordinator. We take this integral of the subordinator, and then this will be our operator. Okay, so this operator will help us in mapping or inherit, sometimes it's called intertwining, because we have the chance to import, in some sense, information on the classical case to the Jacobi with jumps. And of course, if we know the Laplace transform, we can derive the expression and look, for instance, to the first moment of the first passage time. That's useful for the firing rate. But now let's see in a more special case, because if we we cannot say much if we do not say what is the distribution of the jumps. Okay, so we have to specify the distribution of the jumps. Here, we decided to use an exponential type density, okay, for the jumps. If we explicit this pi, we are able to write the generator of the process, and we are able to write the Bernstein function associated to this process, okay, in a closed form. Well, since we have all this information, we can use our theorem. We know the Laplace transform, so we can derive it. We know the first moment of the first passage time. And in this case, we can write it very explicitly. I understand that the formula can look cumbersome, but actually is very easy to evaluate numerically. Okay, so we can evaluate the first passage time. And for instance, what we observe that if we plot the mean first passage time, so the mean time we have to wait before the first crossing of the threshold, decreases if we increase alpha. Who is alpha? Alpha is the parameter of the density of the jumps. Why so? This is just a plot, very simple plot of the density of the exponential density when we change alpha. And we see that if alpha is large, most of the mass will be for small values. Okay, while if lambda is smaller, we will have higher mass for larger values. This means that if we consider alpha large, imagine here, if alpha is large, most of the jumps that are downward jumps will be very small. So if the process is close to the threshold, even if we have small jumps, the small jumps close to the threshold are rare. But if they are also small, we will have that we don't have wait too much before the crossing. Whether if we are close to the threshold and we have a large downward jumps, we are increasing a lot the time before the first spike. Okay, so this is not surprising according to our framework. What is maybe more surprising is this plot here. In this plot, we have the firing rate of the classical Jacobi case. So it's a firing rate as a function of excitatory inputs. Okay, this is the rate of excitatory inputs. So in the case blue solid line, we have the classical Jacobi, and the dashed red line is our Jacobi with jumps. We see that first of all, the firing rate of in our case is always smaller. And okay, this is not surprising because we are introducing inhibition from outside. Okay, we are downward jumps. So we are trying to stop or to decrease the activity of the neuron. So this is something that we wanted. So it's okay, we obtained it. But I want to stress that there's another feature here. If we don't have refractory period, if we consider leaky integrated fire, here it's a Jacobi, but also in other cases, the firing rate increases linearly and unboundedly. Okay, and this is not very reasonable. Whether in our case we have that the firing rate saturates. Okay, so it's you can think that is it's adaptation or whatever you want, but it's true that in this case we observe that a saturation of the firing rate. That's something that's a feature that it's nice. I want to stress that here in this plot I used exponential distribution of the jumps, but this model has high freedom in the choice of the distribution of the jumps. Okay, if you want to model different things, you just have to change the distribution of the jumps. Okay, so there's high degree of freedom. But of course, this is just a first investigation. We need to add upward jumps, and we need to investigate higher moments of the first passage time, and so on, and see how to choose the density in different cases, in different situations we want to model. Okay, so there are many other open problems, but I'm running out of time, I think. So I would like to thank my co-authors, and I would like to thank you for your attention.
Thank you, Giuseppe. Are there questions or comments?
It was a really nice talk.
Thank you, Lindsay.
Questions?
Short question, okay?
Sure.
So is it possible, or why it is difficult to incorporate the excitation instead of inhibition?
Okay, it's very nice.
Difficult technically?
Yes, it's difficult from a mathematical point of view. There are two reasons. One is on the property of this measure that is in the integral. So the jumps, the distribution of the jumps, it needs to have some properties, and also we want that the crossing of the threshold, we want that this for the diffusion, not for the jumps. We want to cross the threshold due to the diffusion, and we don't want to have the hitting time, or we don't want to overcome the threshold with a jump. Okay, so that's the main difficulty, and we are trying to fix it and see how we can
yes, because I think in biologically there is a report about the very large EPSP.
Okay,
so maybe taking into account of a large jump can be also biologically important problem. Just a comment.
Okay, thank you. Yeah, definitely we are planning to. It just from the mathematical point of view, it gave us some issues.
Even approximately, I think it's very important. Thank you.
Thank you.
Yeah, sorry. Ciao Giuseppe. One question I have is you were mentioning that I don't remember anymore. Okay, I need to get that question back.
You're getting old, Massimo.
Yeah. I mean that's a skipping lunch. That's a consequence. Okay. So you were mentioning that you don't want to cross the threshold due to the jumps. What's the reason for that?
Because it's in that case, this expression, I think it's this expression is not exactly this one. We should take into account we have troubles in mainly the technical part of our proof. So we need to cross it in with the diffusion, not with the jumps, because we lose some regularity of all this function of this operator that are involved in the proof.
Okay, because well, I mean, perhaps one can try to condition it on that and distinguish between the two cases, depending on. I mean, if you know the time when the jump occurs, then this might give you some sort of probability confidence on whether you are going to cross for that or not.
Yes. Yeah, definitely. I believe it's possible. I'm not saying it's not possible. Just we have to fix something. But maybe, as you said, there will be two components here or something. We have to distinguish between the two cases, or we can at least start with very small upward jumps. I don't know. We'll still have to fix it.
Okay. Yeah, I mean, if the possibility might be to allow the jumps to have the same amplitude as the possible upward oscillation of the process, right? So if you go from time t to times t+1, and you know that the Jacobi has a possible jump amplitude of that size, and you ask the jump to be of that size, then it might work.
Yeah, actually, it's possible, because here we impose that the downward jump is always smaller than x minus vi. So we never jump below the lower boundary. So maybe we can impose that it never jumps above the threshold, going upwards.
But in any case, we have only downward jumps.
Here, yes.
So you cannot have a crossing due to the jumps.
No, here, yes, but he's saying if we try to extend it.
Okay, to positive jumps.
Yeah.
Okay, I think it's time to close this session. Thank you to all the speakers and to all the participants that I see were very numerous. And now we leave this session, but it starts the mini talks session, and Daniele will take the chair of this part.
Masi, in fact, this is only very briefly to say that channel 2 has closed already, so we can start the microtalk session anytime. So whenever you're ready, we can go ahead.
Yes, so I think that I'll start now, as we are there. Yes. So okay, it's a bit complicated. Okay, let me just do one more thing. I need sorry. Yeah, I need the sorry all. Okay, sorry about that. Let me share again. Okay, the first speaker of this microtalk session is Marcelo Becker. Please, you have 2 minutes from now.
Can you share, Masi?
Okay. I thought I thought of sharing. Am I sharing now? Yes.
Yeah.
Yeah. Okay.
Can you see? Okay. So you have 2 minutes from now.
Okay. Thank you. So our interest in this work was to construct a transfer function that picks up the input of a generic content-based neuron and gives its output rate. Of course, this was done for a variety of neural models, but here we wanted to see if we could deal with the combination of generic nonlinearities in the model and also compound with temporal correlations of the content state current. Our process was to use the diffusion approximation for the Poissonian input and then reduce the dimensionality of the system. This results in a colored and multiplicative SDE, which makes it impossible to construct an exact Fokker-Planck. So our solution to this problem was to use a method derived by Fox to approximate the Fokker-Planck, but we generalize this method to multiple noise sources. And with the Fokker-Planck, we numerically calculate the stationary firing rate of the model. We compared the resulting transfer function with simulations, and from the plots, it can be seen that the approximation describes relatively well the behavior of the firing rate for a large range of parameters of the system. The specific models tested here were the interpolation of a fast and slow excitatory current, and also a model with AMPA and nonlinear and MDA channels. So this method was able to capture the behavior of firing rate for context-based integrated power neurons, and is also in principle generalizable for a variety of other types of models with exponential time correlations. Thank you, and feel free to make any questions in the break room at the end of the session.
Okay, well, impressive. Thanks, Marcelo, for keeping the time. Our next speaker is Maria Virginia Bolelli.
Yeah.
Whenever you're ready.
Hi, everybody. My name is Virginia Bolelli, and I'm a PhD student, and I will talk about neurogeometry of stereo vision, a work that I'm developing together with Giovanna Citti, Alessandra Sarti, and Steven Zucker. What is the stereo problem? The stereo problem is a reconstruction problem of the three-dimensional visual scene, starting from its projection on the two eyes. The main issue is to couple, in a correct way, the correspondent left and right retinal points in order to project them back into the environment space. Our model is inspired by the work of Zucker in 2006, and it's equipped with instrument of differential geometry introduced in the work of Citti and Sarti in 2006. Binocular cells, which are involved in the process, integrate inputs from both eyes through nonlinear interaction of monocular receptive profiles. And since they are sensitive to position and orientation, we decide to model the problem in the space R3 times S2, equipped with differential constraints. These constraints let us choose a suitable distribution D of the tangent space, and integral curves of this structure generalize well to the 3D case, the two-dimensional notion of association fields proposed by Field, Ice, and Dess in 1993 for completion and good continuation. Moreover, integral curves induce a distance and a connectivity curve. And so we can apply spectral analysis techniques to the stationary mean field equation, and in particular, starting from the lifting of the left and right images in the manifold of 3D position and orientation, we see that it's possible that the first eigenvector associated to the mean field equation is able to select the correct 3D points of the stimulus. We can conclude that this special spectral clustering based on the geometry learned from the cortex is able to solve, at least in this simplified setting, the correspondence problem. Thank you very much for your attention, and thanks to the organizers for this nice opportunity.
Thank you a lot, Virginia, for the interesting presentation and for keeping the time. Our third microtalk speaker is Kadmon Laksa.
Hello, are you listening to me?
Yeah.
Right. So I studied the metastable behavior of a stochastic system that is a Galvez-Scherban model with n stochastic neurons and instantaneous leakage of membrane potential. So in this model, at each time t, a neuron A is associated to a membrane potential that will denote ut of A, and will denote ut as the list of membrane potentials at time t. It means that each entry of ut is the membrane potential of a neuron at time t. This model describes the time evolution of this list of membrane potentials as a Markov jump process with the following rates. So if a neuron has null membrane potential, it is not allowed to spike. A neuron with positive membrane potential spikes with a rate that is the exponential of its membrane potential. And each neuron can have a leakage of its membrane potential with a constant rate 1. When a neuron A spikes, its membrane potential is reset to 0, and the membrane potential of the other neurons increased by 1 unit. And when neuron A has the leakage of its membrane potential is resets to 0 with no effect on the other neurons. In this framework, we first prove that with probability 1, the process gets trapped on the list of no membrane potentials. And we also show that when the number of neurons of the system diverges, the rescaled trapping time converges to a min1 exponential random time. It means that when the number of neurons of the system is big, the system gets trapped after a long and unpredictable random time. Thank you. I'm looking forward for questions.
Well, great. Thanks. Yes, perfectly on time. Our next microtalk speaker is Chenggang Chen.
Yeah, I'm here.
There you go.
Hi, everyone. I'm Chenggang. I'm postdoc from Johns Hopkins University. Our lab studying marmoset. It was a new world monkey for 30 years. We recorded the neural activity from auditory cortex. In our extracellular recording showing on the top left panel, you can see example bursting neuron, and we use two metrics to classify bursting and non-bursting neuron. In our intracellular recording showing on the bottom left panel, there are 3 example neuron types. For the bursting neuron, yellow color, it has very narrow spike width and the interspike interval ISI, and the sound stimulation did not change the ISI. For the fast spike neuron, the blue color, it has narrow spike width and likely to be inhibitor neuron, and this neuron has longer ISI. For the regular spike neuron, green color, it has broad spike width and likely to be excitatory neuron, and this neuron has very long ISI. After collecting data from 1,000 neurons, we asked it what is the contribution of bursting to the spiking neural network. So in the previous spiking neural network showing on the top right panel, it has superior performance, but it is difficult to fulfill in a real biological neural network. So in our very simple classification task showing on the bottom right panel, we found that bursting promotes learning in the spiking neural network. Talk with me if you are interested in this work or our data set. There is no other place where you can get in vivo intracellular data from primate. Thank you.
Well, thanks again. Yes, our next speaker is Hong-Gyu Yoon. Please, whenever you're ready.
I've tried to warn this person, but I couldn't see them in the chat.
Okay, let's wait. 5, 4, 3, 2, 1. Okay, we can move to the next microtalk speaker, and then in case Hong-Gyu Yoon show up, they can talk later. Okay. So. And we will jump to Josué Torquanti Fozzo. Sorry for the pronunciation.
Okay, do you hear me?
Not so clearly. If you can increase the volume of your mic, that would be appreciated.
Is it better?
Yeah.
Okay, thank you very much. Good morning, everyone. I will talk about the work in collaboration with my postdoc supervisors, Eva Lockerbach, Patricia Renaud-Bouren, and Etienne.
Sorry, Josué. Again, the mic went down again.
Oh my God, what is happening? Is it okay?
Yeah, now it's better.
Okay. So I was saying I will talk about the work in collaboration with my postdoc supervisors, Eva Lockerbach, Patricia Renaud-Bouren, and Etienne Tanré. Excuse me, I have a problem with my microphone. I will just remove this. Sorry, I have a technical problem.
Now it works. Now it's better.
Okay, is it better now?
Yes.
Okay, thank you. So if I can continue. I was saying, talk about the work with my postdoc supervisors, Eva Lockerbach, Patricia Renaud-Boren, and Etienne Tanry on the detection of neural synchronization. In fact, two neurons are said to be synchronized when the spike trains coincide more than when they are totally independent. And the main question of this work was—
Sorry, Josué, it's still very low. So I'll suggest this. We move to Maxwell Kreider, and then we give another attempt and see if it works better, or in the worst case, you can shout as loud as possible, okay?
Okay, okay.
Okay, thank you.
This happened with this.
Okay, hi everybody, I'm Max Kreider. I'm a grad student at Case Western Reserve University. So let me jump right in. There are different methods to quantify the synchronization of systems of coupled deterministic oscillators, which often rely on a phase reduction-based approach. However, it can be a challenge to extend these methods to systems with stochastic oscillations. So here we study a model of two Morse-Laccar neurons with channel noise. This is characterized by noise strength epsilon, which can be interpreted in terms of the total number of ion channels of the system. We also have gap junction coupling in the voltage dynamics, which is characterized by coupling strength gamma. Now, in order to quantify the synchrony of our system, we utilize the notion of the power dissipation through the gap junction, which is given simply by the product of the coupling strength and the mean square voltage difference. We note that the power dissipation has a physically meaningful interpretation, simply the energy per unit time required to maintain the system in the synchronized state. In order to obtain analytic results, we approximate the evolution of the voltage difference as an Ornstein-Uhlenbeck process, where we derive the effective noise term DT from first principles by treating the ion channels as binomially distributed. To further simplify the dynamics, we use an averaging argument to arrive at a constant coefficient Ornstein-Uhlenbeck process approximation. And we're hypothesizing that the power dissipation can be well approximated by the product of the coupling strength and the resulting stationary variance. And then finally, our results show that there's excellent agreement between our theoretical prediction and the numerical simulations, and indeed that the power dissipation can be characterized by a quadratic dependence on the noise parameter epsilon. Thank you so much for your attention.
Thanks a lot, Max. As usual, everyone can then have the time for more question discussion in the breakout room. Our next speaker is Benoit Duchel.
Thank you so much. So modeling spike timing dependent plasticity, or STDP, is key to understand how neural networks change over long time scales. However, simulating neural networks with STDP is computationally very costly, and there's no low dimensional description. So the goal of this work is to build mean field approximations of adaptive networks with STDP. In STDP, when either the pre or post-synaptic neuron spike, the connection between them is depressed or potentiated depending on the difference in spike timing. Perhaps the most famous form of STDP as an anti-symmetric kernel, but here we focus on symmetric STDP. To approximate STDP in networks of phase oscillators, it is convenient to consider simpler phase difference dependent plasticity, or PDDP. These rules are based on phase differences rather than spike timing differences. We also propose event-based PDDP rules, or EBPDDP, which are conceptually closer to STDP. Turns out that single harmonic PDDP rules can approximate a simple form of symmetric STDP in terms of the coupling, so that's panels C, D, and E, as well as synchrony, and that's panels F and G. We derive exact expressions for the evolution of the average coupling in terms of network synchrony, and use this to approximate Kuramoto networks with STDP that form clusters. So in panel D, we can see two dominant clusters. And if we assume that the couplings within and between clusters are homogeneous, we can write systems of ODEs for the evolution of the order parameter and the average coupling. Finally, we show that the two-cluster mean field model can be fitted to provide a low-dimensional approximation of the full adaptive network with symmetric STDP, and that's panels H to L. Many thanks to my collaborators, Chris and Anya, and I'll be presenting more details in the breakout room for those interested.
Thanks a lot, Benoit, spot on. Our next speaker is Miriam De Graaf. Please, whenever you're ready, unmute yourself.
Okay. Hi everyone, I'm Miriam de Graaf. I'm a PhD student at the Department of Movement Science of the VWU Münster, and I'll tell you a little bit about how motor pattern generation is robust to neural network anatomical imbalance favoring inhibition, but not excitation. So studies have shown that functional balance between excitation and inhibition is of course critical for producing motor patterns. Despite this functional balance, biological neural networks have an underlying anatomical imbalance, for instance, in a number of excitatory and inhibitory neurons. Reservoir computing neural networks, however, have a functional balance that is imposed via an anatomical balance. This made me wonder, what is the influence of introducing anatomical imbalance in reservoir computing neural networks? So to investigate this, I created a reservoir computing neural network with separate pools of excitatory and inhibitory neurons. I then varied the excitation to inhibition ratio in neuron number, connection percentage, and connection strength separately. I trained the networks to reproduce locomotor muscle activations that were estimated from kinematics via inverse dynamics calculations for the muscles of the S1 spinal cord segment, in such a way that the reservoir kind of becomes a sort of abstract model for the spinal cord segment. Now, I found that when networks are imbalanced towards inhibition, which you can see on the left side of the two plots, that we see a sufficient number of principal components in a network activity, which was accompanied by, in the bottom figure, a high performance. Now, as imbalance shifts towards too much excitation and we move the right of the two plots, we see in the top plot a drop in the number of principal components that is caused by a saturation of the neural firing rates, which is accompanied by a very sudden drop in the performance that we can see in the bottom figure. So we concluded that network performance relies on network dynamics having sufficient richness and something that I've actually not shown here, also sufficient structure, which we lost when the connection strength was too high. And our main finding is that network performance is robust to increased inhibition, but not to increased excitation. Thank you for your attention.
Thanks a lot, Miriam. Our next microtalk speaker is Simone or Simon Ebert. I'm not sure. Sorry about the pronunciation.
Okay. Hey, thanks. You can hear me all right?
Yeah.
Okay, perfect. So hello, everyone. Thank you very much for letting me present my PhD work here, which is on the role of dynamical synapses in retinal surprise coding. So at the first stage of visual perception, the retina efficiently encodes visual input into spike trains that carry relevant information to the rest of the brain. To this end, a long-standing hypothesis is that it does so by mainly responding to changes rather than the raw image itself. You can see an example of this behavior here in the response of a retinal ganglion cell, which, when presented with a regular sequence of flashes, does not respond to the repetitive stimulus itself, but rather emits a big error signal when this pattern unexpectedly ends. And what is even more intricate is that the time point of this response scales linearly with the period of the flash sequence, indicating that the cell had a temporal prediction of when the next flash should have occurred. So based on experimental findings of our collaborators, we designed a simple computational model of the retinal network in which dynamic inhibitory synapses that undergo short-term plasticity enable the retina to emit a precisely timed error signal. So via an additional inhibitory input in the network, the time point of the response peak, which signals the offset of the stimulus, is delayed. And via short-term plasticity, the weight of this input adapts to the frequency of the periodic flashes. As a result, the synapse depresses for high-frequency flashes and thus provides little inhibition, whereas for low-frequency flashes, it maintains a higher weight, thus pushes the response peak further back. With this work, we show how dynamic adaptation can enable the retina to temporarily refine its prediction and send precise surprise signals when these are not matched. Overall, this shows that the retina provides highly sophisticated inputs into cortical processing, and in the future, we want to explore how short-term plasticity in a larger network may enable the retina to extend this sophistication to more realistic spatiotemporal stimuli. Thank you very much for your attention, and that was it.
Thanks a lot. Josué, should we give it another try?
Yes, I hope, I think it's okay now.
Okay, yeah, we can hear you.
Okay, thank you. I will talk about a work with my postdoc supervisors, Eva Lockerbach, Patricia Reynaud-Bourret, and Etienne Tanré on the detection of neural synchronization. In fact, two neurons are said to be synchronized when the spike trains coincide more than when they are totally independent. And the main question of this work is how do we detect it given an IIG sample of pairs of spike trains. In order to answer this question, we developed a statistical test for which we formulate the problem as a test of independence with the U statistic given by UN that compares the coincidence given a delay delta of the spike trains of both neurons under the same observation and the coincidence under independency. For this kind of test, the main difficulty is that we don't know in the non-asymptotic framework the real law of the U statistic. So we showed that we are still able, through a specific criterion, to allow the test to have the good control on the first and the second species risk. And we also did some applications on the case of Poisson point process for the modelization of the spike trains and the case of Poisson point process and shifted Poisson point process. And I think we can discuss a bit more about it in the breakout rooms. Thank you.
Perfect. Thank you, Josue. Thank you all. Let me try again to call Hongyu in case he's here. Last attempts. And there we go. Yeah. No, that's Etienne talking. Yes, okay. Okay, then I think that the breakout room are now open. So just feel free to join the room that you prefer. Very simply, you just have to go here, breakout rooms, you join the one you prefer and enjoy that. Thanks, everyone.
Ma sì, mi senti? Posso dire due cose?
Yes.
Lo dico in italiano perché interessa soltanto a noi due. Ma guarda, io sono molto impressionato con tutti questi giovani che hanno fatto delle presentazioni. Tutti.
Only a few people can understand you, but those who can really appreciate that you like young people, Antonio.
No, it was very, no, Daniele, no, questa è una cosa dell'organizzazione, ma è vero, sono molto impressionato, è gente interessante che fa le cose. Allora penso che dobbiamo invitare
Antonio, you are on YouTube and the language of this conference is not Italian, it's English, I'm afraid.
No, ok, Massimiliano, ti voglio parlare dopo, va bene?
That's fine. That's fine.
Suan, are you around?
We can't hear you at the moment.
Can you hear me now? Sorry, I had my.
But it's quite quiet.
Okay, let me just. Yeah, I had it turned off. 1 second. How's this? Is this better?
Yeah, that's better.
Okay, yeah, I had to switch it to my other microphone. Zoom always picks the wrong one, right?
Daniele, I just noticed that we're still recording. I don't know if you wanted to stop it or okay.
I think that for practical reasons, I left the YouTube on and also the recording on, and then I'll do the right trimmings.
Change, yeah. Okay.
Yeah.
So, Suan, do you want to check your?
Yeah, let me just see how this works.
well, you think, is that, you're gonna have to scroll through the slides there, or is it? I think you're on. Yeah, that looks. I think you're on mute there. Can't hear you.
How's that?
Yeah, that looks fine. We can still see kind of the outline, but it's no big deal. You can leave it up there. Easier.
Okay, let me, I'll try one other way. Sorry, I've been having Zoom issues recently. How's that?
Yeah, that's better.
Okay, that's better. I can't see you guys while I'm doing it, but that's maybe that's better. So, yeah. Yeah, I'm running Zoom through my browser because the app's not working well for me. Okay, I will do it that way. Also, it doesn't seem to mess up my microphone as much. Okay, thank you so much. I just wanted to try all these different options.
While I have you, in terms of questions, if there are questions during the talk, would you want to allow people to ask questions during the talk, or would you prefer to have them all at the end?
I can allow them. I won't be able to see the chat because I'm going to be in full screen, but if someone wants to like read it to me or something, that would.
I'll moderate the chat, and if there's a question that seems like it's pressing to ask at the time, I'll ask it then, and if not, I'll ask it at the end.
Okay, sure. Yeah, I have no problem answering during the talk.
Perfect.
Okay, so I guess we're all set.
Yeah, so we're starting back at, well, in 35 minutes time.
At 35 minutes. Okay, I will see you in a few minutes.
Okay.
Bye.
Bye.
Okay, we'll get Joanne's.
Daniele, do you think we should maybe close the breakout rooms? Bring everyone back in?
I was just writing that in the chat, so I'll do that right away.
we'll give it a couple of minutes for everyone to filter back in. But maybe, Sivan, if you want to start sharing your screen so that it's up when people come in.
Okay, can you hear me okay?
Yep.
Okay, and I won't go to full screen just yet for my talk, but when I start, I will. Okay.
We'll wait another minute or two.
Okay, great.
Okay, so we'll get started. So welcome everybody to our final of this year's ICMS conference. It's my pleasure and privilege to be introducing Professor Sue-Ann Campbell, who's a professor in the Department of Applied Mathematics at the University of Waterloo. So Sue-Ann obtained her PhD in theoretical and applied mechanics from Cornell University, after which she went to the University of Montreal as postdoc, and she had a short stint as an assistant professor at the Concordia University before returning to Waterloo, which is where she is now. In her research, she develops and analyzes mathematical models of neural dynamics. She studies such dynamics on a range of different spatial scales, from single cell all the way up to population level. And a major focus of her work is the role of nonlinearities and time delays in shaping these dynamics. So today, Sue-Ann is going to talk to us about modulating synchronization with a slowly building ionic current. And if you have any questions during the talk, please put them into the chat, and I will pose them to Sue-Ann if the time is appropriate. And then non-pressing questions will be reserved for the discussion after the talk. Over to you.
Okay, so can you see my slides okay?
Perfect.
Okay, so I just wanted to thank the organizers for the invitation to speak. It's a great honor to speak here. And actually, as the last speaker, I wanted to, on behalf of all the participants, thank the organizers for a fantastic conference. It's I've enjoyed it very much. I think it's been very well organized, and the time zones worked out really nicely for many different people. So thank you very much for that. Okay, so let me get started. First, I want to thank my collaborators on this project. So Issam Aldarabsa was a postdoc that working with me for a few years, and now he's in the Department of Mathematics and Statistics at University of Manitoba. And he did some of the work on the first part of the talk, and then the second part is work with Victoria Booth at University of Michigan and Shuying Wang at Washington State University. And I want to acknowledge the support of the American Institute of Mathematics. So this work was really instituted when the three of us participated in Research Square. We're only three of us, we were a triangle, but at AIM, and that really helped us to kind of get the work going. And also my usual funding agencies and the university that helped support Issam's postdoc. So just a little bit of background. The work I'm going to be talking about today is related to neuromodulators. So neuromodulators are chemicals that are released by neurons that can modulate the network behavior in a neural network by affecting synaptic transmission or by affecting the intrinsic excitability of neurons. So the neurons can release neuromodulators. They might affect a neuron, or it might affect the synapses. Today we're going to be thinking about the one I'm interested in is acetylcholine, which is thought to play a role in arousal, attention, memory, learning. On a smaller scale, it can affect the frequency and coherence of cortical oscillations. So the word I'm going to think about is where it's modulating the neuron, the effect of a neuron. So here's some very old experimental evidence looking at particular neural cell. I believe it's a pyramidal cell, and this is under control, and this is under the effect of acetylcholine. And what you can see is it's changing the spiking behavior. And we've heard a lot in the last few days, at least a lot of talks I went to, we're talking about spike frequency adaptation, and you can see how this acetylcholine is modulating that spike frequency adaptation. So here's a quite strong adaptation. So the spike frequency adaptation is when the spiking time changes over the course of the period of stimulation. And here you can see it slows down and actually stops. And here there's a change in the interspike interval, but it's not quite as strong. So we go from kind of strong spike frequency adaptation to weak. And so one of the ways it acts, many different ways, but one of the ways it acts is through potassium currents. So in a particular one, the muscarinic potassium current, the acetylcholine acts in a particular receptor, the muscarinic receptor. So a number of people have studied these effects in neurons. So some of the old work is like this. Some newer work has looked at, so Stifel and Al looked at phase resetting curves of neurons and how acetylcholine affect that. Prescott looked at resonance effects. So there's many different ways that acetylcholine, we observe that affects the spiking or excitability behavior of individual neurons. And then at the network level, this is some old work that looked at rhythms in the hippocampus, looking at in CA3 and CA1. And in this case, they use carbachol, which is basically acts like acetylcholine, mimics acetylcholine effect on the IM current. And so you can see these sort of large rhythms arise, and when they shut down, essentially block the receptors, and the rhythms go away. So, so you can see that this had a very large effect on the network behavior. And more recent, really interesting work, acetylcholine has been associated with changes in rhythms during sleep. So here they're measuring in simultaneously EEG and EMG in rats, but they're also measuring, these units are neurons that secrete acetylcholine. So there's a kind of a correlate. This is the wake, slow wave sleep, and this is essentially REM sleep. And there's different, you can see as there's different sort of rhythms happen, you can see also changes happening at the same time as acetylcholine. So that definitely seemed associated with that. But the really neat thing about this study is that they actually showed more than just a correlation, but that the fact that acetylcholine induces the rhythms because they used they were able to use, the word is leaving me right now, they used genetically engineered rats where they could turn on the production of acetylcholine by these particular neurons by light. Okay, and so they could turn on the production of acetylcholine, and they could see sort of changes from kind of a stronger rhythm to something kind of smaller afterwards. Okay, and they looked at power, changes as power and things like that. So this is really neat because they could show it wasn't just sort of they happened at the same time, but it really seemed that the acetylcholine was causing this change in the rhythm. Okay, so getting to like what were we trying to do with this work? It seems acetylcholine has these different these effects at the single cell level, and that they may scale up into this effect at the network level, but they tend to vary more slowly over time. So we were interested in how this slow variation may affect the behavior in networks. In particular, we thought about thinking about synchronization. So whenever you see these kind of large amplitude oscillations coming out of the network level, you think of synchronization. So this is going to be two-part. The first part, I'm going to talk about some work that I did with the SOM, just looking at the effect of a static effect of acetylcholine on single cells, and just kind of get a kind of general picture of what it does from a kind of bifurcation analysis point of view. And then the second part, I'll really get into thinking of the slowly varying acetylcholine. Okay, so just to set the scene, so I'm really small. This talk is going to be pretty small, single neurons and small networks. So a standard conductance-based model for neuron voltage, neural excitability, such as the Hutching-Huxley model, looks something like this, right? So we have an equation for the voltage that's varying in time. So V is the voltage across the cell membrane, right? We have the capacitance, standard and applied current, which as you vary that, it'll change the spiking behavior and spiking frequency of the neuron. And I sort of rolled all into this variable here, these ionic currents, which are typically associated with producing the action potential. So in the Hodgkin-Huxley, it would be sodium and potassium and lead current. And then associated with those are gating variables, which typically have the form equations in this form. So if we look at the variation, as I mentioned, of I applied, it's quite well known that as you increase the I applied in those models, it mimics what we see in many neural behavior, that you get oscillations arising, and there's kind of a classic way of classifying how these oscillations arise. So originally done by Hodgkin in 1948, you can see as the oscillations start, this is plotting frequency versus I applied. You could have them sort of come in at zero frequency and gradually change, or you could have them come in with sort of a jump in frequency. So Hodgkin did this work in, I think, Crab axons or originally, but then in 1998, Rinzel and Ehrmantraut looked at how this could be related to bifurcations in models of conductance-based models, as I just mentioned. So the class 1 is typically associated with the saddle node and invariant circle bifurcation, or it can also be with the homoclinic bifurcation. And the class 2 is typically associated with the fold limit cycles and a subcritical Hopf. And there also has been work which we'll be talking about how that type of excitability may affect how the neurons synchronize when they're put together in a network. But first I want to mention some other interesting work. So if you choose, so in the last plots I had I applied as my bifurcation parameter, and if I take some other parameter in the model and do what's called a 2-parameter bifurcation diagram, it has been shown in many different models using different things as this different parameters, so such as leak conductance or potassium conductance, different, that you get a kind of, you seem to see the same bifurcation diagram over and over again. And I'll just explain a little bit. So the green lines here are fold bifurcation. So this is where equilibria are coming together and disappearing. This is a cusp point where those two come together. The blue line is a line of Hopf bifurcation. So as we cross here, we get the periodic solutions appearing, and this is double. So this is two code-mentioned 2 points, the cusp point here and the Bogdanov Hawkins point. So where the Hopf comes in and sort of ends at the fold bifurcation. And so this was observed numerically, and it's in Bart Ehrmantraut and Dave Terman's book, this kind of bifurcation diagram. We see it a lot in various models. And then in 2015, there were 2 really nice papers who looked at, how generic is this for, because I just wrote down a sort of a generic conductance-based model, and they looked at sort of how generic is this bifurcation for an arbitrary conductance-based model if we don't put specific, currents in there, but we have the kind of standard currents in there. And so one of the things we see with this is that if we think of now whatever this parameter is, fixing it down sort of low, we get class 2 type of oscillator as we vary I applied. But if we move it up and we then vary our I applied up here, then we get that class 1 down below and class 2 up above. So varying this up per other parameter, whatever it happens to be, can modify your model from being a class 1 to a class 2 oscillator. So Issam and I were motivated by this work and by what's been observed in the work it's observed in neurons that have the M current, to think about whether this kind of bifurcation, so it has been observed in models with the M current, is to see if it's whether it's sort of generic for that. So what we looked at was it's a general conductance-based model. So same things I had before, but now we specifically make sure that there's this IM current in there. So the IM current's a potassium current. It looks something like this. We have this is a constant maximal conductance. There's a gating variable M. Okay, this is a typical equation for that, and it has a reversal potential VK, which is potassium. And what we'll be thinking about is that the effect of acetylcholine is to downregulate, so turn off this channel. So when acetylcholine goes up, the GM will go down. So we're thinking of this as being another parameter that we want to think about varying in the model. The models for IM, so how do you choose the tau M and the M infinity? They've been fit for various different data for various different types of cells. So the M current occurs in interneurons. It also occurs in hippocampus and cortical pyramidal cells. So it's very, very much all over the place in the brain. So building on the work that I mentioned of Kirst et al. And Pereira et al., what we did, I'm not going to go into details, but we looked at this as a sort of general model and did some basically normal form and bifurcation analysis. So I don't want to go into details. You can show equilibrium points. The key point in getting an equilibrium points is they have the M variable and the WI variable have some specific form coming from their equation. And then we have this function that comes from the right-hand side of the voltage equation. It turns out that's quite important in determining sort of what happens in the model. And so, for instance, you can show that the kind of known properties of these functions and the positive parameters. You can show there's always at least one equilibrium point for any set of parameters. But more interestingly, you can formulate conditions. So these two codimension 2 points that I showed in the sort of generic model that we see in many different neural models, we can formulate conditions on when those will exist because they seem to be acting as organizing centers for the behavior of these models. So again, lots of sort of math swept under the rug, but you have these details. You can say if you want to have a Bogdanov-Takens bifurcation point, then you have certain conditions that need to be satisfied. And what we can show is that they can be satisfied by choosing, so we need some parameters to vary in our normal form by choosing the applied current and the maximal conductance of the M current as those two parameters. So this will give you the Bogdanov-Takens bifurcation point, and similarly, you can formulate conditions for a cusp bifurcation point, and you can show those can be satisfied by varying these two. So essentially, this M current together with the applied current can induce these two codimension 2 points, which give that kind of generic bifurcation structure. And you can go on from this actually and look at codimension 3 points, but I don't want to go into that. So here's some examples. This is the two that I mentioned from before, the cortical parameter cell that Stiefel et al. Looked at, and this is the hippocampal pyramidal cell looked at by Olufsen and et al. And all of these plots I have IAP on this parameter on this axis, and GM, the conductance of the M current, on the vertical axis. And then here's one we just created. So we took the standard inhibitory interneuron of Wang-Buzaki and added the M current. And I think we just took the M current from the Stiefel model and maybe just and threw it in there. And so the interesting thing is that in all cases you see this so what the prediction was, right, we should get these two codimension 2 points, the Bogdanov, the cusp, in all cases, and they do organize, and we see the sort of same backbone structure of bifurcations. So I said we had the fold, the homoclinic, and the Hopf. And the implications I mentioned before, but just to remind you, if we're in the single cell, this is just to show you from the Stiefel model, right? If GM is below the BT, so we're kind of down here, then as we vary I applied, if I produce as I cross here, I have a subtle node on an invariant circle bifurcation. So this is a one parameter bifurcation of voltage versus IAP, and this is just showing as I vary I apply. So here I get periodic solutions, and this is showing the frequency. So I get a class 1 oscillator bifurcation as I go across here. And then when I get above that Bogdanov-Takens point, I get a class 2 oscillator. And there's actually a fairly complicated sequence of bifurcations that we go through to get to that, but I'm not going to go into that much. But the key point here is that we have sort of this generic bifurcation structure that we see in all of these models with the M current. And then by varying the M current, we can switch the system from a class 1 to a class 2 oscillator. And so this is kind of well known when you think about class 1 and class 2 oscillators. Now what happens when we couple them together? So if we look at just 2 cells, then, and with inhibitory coupling, when we're in less than this BT, so we're in the class 1, we get synchronized behaviors. This is just coupling them together with standard inhibitory synaptic coupling. And if it's large enough, then we get desynchronized. So here we get antiphase behavior. So this is all with the pyramidal cell from the Stifel model, but we see the same thing if we do the other models. And this is kind of a busy slide, but just trying to illustrate, like we see that. So what I'm showing here is as we vary GM, I have 2 coupled cells, and I'm looking at are the cells basically synchronized or not? And we're looking at the phase shift, phase difference between the 2 cells. So this is excitatory coupling, and this is inhibitory coupling. And these are the 3 example models that we looked at. And they're not exactly the same, but we see very similar bifurcation diagrams. When the GM is low, in the excitatory case, we have antiphase coupling, and in the inhibitory case, we end up antiphase behavior. In the inhibitory case, they're synchronized or in phase. And then as we change the GM, we get higher. We have some sort of in-between behavior. This is they're synchronized, but the phase difference is somewhere between pi and 0, or half a period and and 0. And then eventually we get to synchronized behavior. So in the excitatory coupling, and the inhibitory sort of has the opposite behavior. Not so clear. This is all done with numerics, so maybe not so clear with the bifurcations happening here. And of course, we see some, for larger things, we see some other different behaviors. So this is the Wong-Buzaki. But the idea is we have this sort of same backbone structure with the 2 coupled cells when we vary this this conductance of this M current, which again, we're thinking about as something that acetylcholine can affect. Okay, and I think I just have one little, before we move on to the second part, little illustration of this, right? So this is in a network of 10 cells with inhibitory coupling. And so as we sort of expected from the 2 cells, if GM is small, then things are synchronized. And so if we look at sort of, this is just looking at the average voltage, right? So kind of to a very simple metric of network behavior. Then we see this kind of large amplitude oscillations, whereas if GM is large enough, they desynchronize. And if we look at kind of the network average, we see this low amplitude behavior. So something that maybe is not so apparent at the, if we measure something at the network level. So if we compare to that old paper, right, where they had the large amplitude, sort of what we think is synchronous behavior occurring when the acetylcholine was acting, right, then the theory would say that that would mean that it would be due to inhibitory coupling, right? Inhibitory would get synchronization when the acetylcholine is on and not when it's off. And the experiment, this is sort of retroactive model verification. The experiment actually showed that blocking the inhibitory synapses destroys the rhythm. So it's self-consistent. It does make sense with with what the theory is predicting. Okay, so that's the summary part. Part 1, right? Just looking at the static effect of this M current. From a bifurcation point of view, we showed that using the maximal conductance of this M current and the standard I apply as bifurcation parameters, you can get these 2 codimension 2 points, which really generate the bifurcation structure of the model. And if the BT bifurcation is quite important in terms of switching how oscillations are produced in the model when the I applied current is changed. So depending on whether GM is above or below that BT, we can have class 1 or class 2 oscillations. So the idea would be the acetylcholine could, which decreases GM, can switch the model from class 1 or class 2, from class 2 to class 1 excitability. And as we showed, associated with the switch is we expect a change in synchronization behavior when the 2 or more of the neurons are coupled together. Okay, so this stuff appeared in a paper in Journal of Math Neuroscience last year. So the second part, right, now we want to think about, we have that sort of structure and understanding of how acetylcholine works in a static case. We want to think about what happens when it's slowly varying over time. And we're going to focus in on how it affects the synchronization. So in the case when the neurons are oscillating, there's of course lots of other things that you could do, but that was the focus for this work. So we're going to take that model that we had, and now sort of we have a generic model, and now we'll think about coupling the neurons together with, you know, in a network with some synaptic currents. And I think I just have, yeah, so this is just sort of standard synaptic gating with a gating variable. And this is what I used in those simulations before with the 10 neurons or 2 neurons. So we have a gating variable, we have a maximal synaptic connectance, we have a reversal potential, which will determine whether it's excitatory or inhibitory, and we have some connectivity matrix. So for most of our analysis, we're actually going to focus on 2 neurons, because that gives us, as we saw with those little simulations I did before, that gives you at least a general idea of what's happening. So because we're thinking about oscillatory neurons, what we're going to do is use phase model theory. So I'm going to try and have lots of references down here, but this is, you know, pretty well-known standard work, right? If I have a system of weakly coupled, and I'm going to focus just on identical for now, oscillators. So if without epsilon is representing the coupling, with no coupling, each neuron would be just oscillating on its own. And I'm going to allow for a parameter in here, because eventually that's going to represent my my GM in my particular current. And for some range of values, each in the uncoupled system has an asymptotically stable periodic solution. Okay, and then we think about what happens when you couple them together. And this theory tells us if the coupling's weak, so epsilon is a small parameter, then you can map these solutions to this phase model. So now we have one variable for each oscillator or each neuron, which just represents the phase, right? So what this model is going to tell us about is the relative phase of these oscillators. So these will not tell us about the destruction of oscillations. We're assuming the oscillations are there and that the coupling doesn't destroy the oscillations, it just changes it in some way. And so the neurons are synchronized or not. And so this phase model will tell us how the coupling affects that synchronization. So we have some frequency here that depends on this parameter. And of course, the coupling, when we do the mapping, also depends on parameter. And you can map out, as beautifully discussed in both of these books, how to actually calculate this. You can map it out. You can do it numerically. And we did that for some models using XVP dot. So now what we're interested in is the parameter not just being fixed, but being slowly varying. Okay, so I'm going to put a delta in there, and the idea is we have sort of 2 small parameters. We have the weak coupling, which gives us in its own a sort of small time scale, and then we have a slowly varying parameter. And so Young and Park in 2016 had a lovely paper where they looked at the case when these 2 timescales are the same, so delta is equal to epsilon, and beautiful work. And so we kind of built on their work because we were interested in actually even slower timescale, so that when so we'll go to a slow timescale later on, that we still sort of see the effect of this slowly varying parameter. Okay, so when we do that, and I'm again not going to go through the sort of hiding all the math, you can again get a phase model. Here I've put it in terms of the slow time, and we have this, and I've, sorry, forgot the frequency, but the frequency is going to disappear in a minute anyways. The point is you have this, essentially when you go through the work and all the perturbation theory, you can show essentially we have the same coupling function we had before. We have now the slowly varying parameter in there, right? That's going to affect, you know, the form of this this function, which again, we have to calculate through some integrals and that. But we have now a phase model with a slowly varying effect in it. So what we want to do is think about that and how that affects our solution. So as I said, just to illustrate, I'm going to think about what happens if we have 2 neurons, because it's really easy to visualize and see what happens. So if we have 2 neurons and the M current is fixed, the GM is fixed, right? Then we're just in the sort of the first case, we have our frequencies and the coupling functions, and you can show it just depends on when they're identical neurons, depends on just their differences, these H functions. So you can actually formulate a single equation for the phase difference phi in terms of the slow time. And we get this nice little equation where h is an odd function of phi. It depends on the parameter. And you can, as I said, you can calculate this given a particular system. So, okay, yes. So what do we do with that, right? If this is just a one-dimensional equation, so you can find equilibrium points, or you can have your second year differential equation class do this. Calculate the equilibrium points by keeping in mind what's behind all this. These equilibrium points of this one-dimensional equation correspond to phase-locked solutions of the original full model. So we have two neurons, right? And this is telling us, and they're oscillating, and these equilibrium points are going to tell us they would correspond to a particular phase-locked solutions where they're oscillating with some particular phase difference given by phi star. And they're asymptotically stable if h prime at phi star is bigger than 0. So as I mentioned, you can calculate the h for given a model of your system. And so we did this. This is actually for the Stifel model. And you can see compute what the h function looks like, and then we looked statically as we varied the GM, how does that h function change? So you can see, again, it's a one-dimensional equation. You can do everything graphically. You can see there's always equilibrium points at 0 and pi. So they always have the in-phase and anti-phase solutions, which just comes down to from the original structure of the model with two identical coupled neurons. But as we vary the GM, the stability changes. This is the excitatory coupling case. So it goes from the antiphase being stable, that's right, antiphase being stable with low GM to in-phase being stable with high enough GM. And you can see this kind of smooth change. And in between, we have these other equilibrium points that appear and disappear, and that causes where we have a sort of transition between the different states. So I think I have summarized over here. So if the M current is low, GM is small enough some value, you get antiphase behavior. In the middle, there's some other phase lock behavior. And when it's high, you get in-phase behavior. And you can map this out in a very crude bifurcation diagram if we look at the phase as a function of GM. And this should be reminiscent of those pictures that I showed before using simulations. And now we're doing it just with our phase model. So those were simulations of the full two coupled neurons and just looking at the solutions. So they were just showing the stable behavior. And here with the phase model, we can actually map out the solutions and look at their stability. And so we have, again, excitatory coupling, low GM, we have the antiphase behavior that's stable, and then we have a pitchfork bifurcation, right? Giving rise to actually two other stable phase-locked behaviors with different phase difference between 0 and pi, or pi and 2 pi. And then as you increase GM further, you come back, and if you think about identifying 0 and pi, this is just another pitchfork bifurcation, sort of a reverse pitchfork bifurcation, where we, and then we go, the synchronous behavior becomes stable. So this is a kind of actually a really common bifurcation diagram that's seen when you're varying stability. But we're interested in, of course, what happens if the current's slowly varying, right? So this was our phase model with the slowly varying parameter. So if GM is slowly varying, right, now instead of being this representing static behavior for different values of GM, now we have to think about slowly moving through this bifurcation diagram. So it's a problem in slow passes through a bifurcation. In fact, if we're going from here to here, or we'll also think about going the other way, because in the system, the CG:1 goes up and down, and the CGM should be going up and down. Then we have slow passage through two pitchfork bifurcations. So we want to think about what's that going to do in our system. So there's some nice work about this. We took a kind of simple approach that really, this is like really beautiful work that looks at a lot of details. We look at kind of a simple, there's kind of a basic thing that you can see that comes out of it. And we looked at a simple way of expressing that also gives a way of understanding the behavior in a different way, let's say. So if we have these pitchfork bifurcations, if we're close to the pitchfork bifurcation, then we can write down a normal form for that, right? So if X is just whatever your amplitude in our case, think of it as like, say, the the phase difference in the model. Then we could write down this normal form of the pitchfork bifurcation with slowly varying coefficients, right? So if we think of taking that phase model and putting it into the normal form, we expect both of these coefficients to end up being slowly varying. And so if we put that in another slow time, so now we pulled this epsilon out front, then we get something like this. This is again, this is something that your second year differential equation students can probably solve, just a Bernoulli equation, so we can write down an exact solution, but we have this parameter in there, and when we write it this way, we can write down the exact solution, and we have this 1/, epsilon, and that allows us to do some asymptotic analysis and try and think a little bit about what happens. So we're thinking about what happens as we pass through the pitchfork bifurcation. So again, I won't go into too many details. An important sort of thing here will be as we vary, so we're varying a parameter in time, right? So as we pass through here, the sort of integral of this lambda of s is going to be important. So the bottom line, I'll just show you a couple of results. So you can do asymptotic analysis, and this gives a lot of details, but the bottom line is if you start from the trivial equilibrium and you pass through the pitchfork, so we're going to have to think about going both ways and also think about subcritical and supercritical pitchfork bifurcations. What happens is if you were doing it statically, you have a static bifurcation point. There's a particular value of lambda where the bifurcation would occur, and we could think about at what time that would occur, right? So there's a time where that occurs. When you slowly vary, and this is seen in slow variation through Hopf bifurcation is probably more well known, but it's the same idea. Essentially, you get trapped close to that stable equilibrium point, and the bifurcation gets delayed. And so you don't see the transition until a later value of lambda corresponding to a later, and so sort of a later value in time as you're slowly varying through the parameter, the bifurcation diagram. And you can do the same analysis with a subcritical Hopf, and you actually see a very similar thing. So you see this delay in bifurcation, and that's going to be really important when we think about, what's going to happen with the slow variation. Now, what happens if we do the opposite? So people don't always think about this, but we need to think about this in this problem because we're thinking going back and forth, and also because of the structure of the two pitchfork bifurcations and how they occur. What happens if we start on the non-trivial equilibrium and then we go into the pitchfork bifurcation from that direction, as opposed to starting from the trivial solution and branching out? And so you can do the same asymptotic analysis, and what you see, again, I won't worry too much about details, is basically we don't have this bifurcation delay. So the prediction from the asymptotic analysis is that you should just slowly follow those non-trivial equilibrium points into the trivial equilibrium point. Okay, so that was just sort of thinking about the pitchfork bifurcation normal form and with a slow parameter. So if we have, of course, have this phase model, and so what we're thinking of is that if we're close, we can think about what's happening sort of close to the two pitchfork bifurcations that I pointed out. So if we're near the pitchfork bifurcation of the zero equilibrium point, we could do an expansion, basically write this in the normal form, introduce a slow time, and then we have the same setup. And of course, we could do the same thing. So we could do sort of have what's happening near the zero pitchfork bifurcation of the zero equilibrium and the pi, right? So we have both of them. And then try and sort of patch these things together. So we have the predictions of the asymptotic analysis, but then we also have that exact solution for the variation of this. So what I'm going to show you next. Oh yeah, and so this is, so to make this a little bit more concrete, we tried some different things, but you need to think about how these parameters are varying with GM, and then of course, how is GM varying in time? So we took everything as just linear. This is probably not a bad assumption when we're close to the bifurcation point. And here, just took a linear ramp in time. We tried different things, but just for the point of illustration, we'll play with that one. And when you do this and you sort of map through all the stuff, what it shows is that if you think about, what is that, how does the bifurcation delay manifest itself, then the delay, how we're going to not see the bifurcation when we're supposed to, but it's going to occur at a later value of the parameter. And what you can show is that the amount it's delayed, so how far along you'll be before you see the bifurcation, is equal to how far you were when you started, how far away from the bifurcation point, which kind of makes sense because you're sort of spending more time close to this equilibrium point, the stable equilibrium point, and getting stuck there longer. But for this linear stuff, it does not depend. It should not depend on the ramp speed. Okay. Okay, and then we can write down the exact solution in that case. Good. Okay, so we have about 10 minutes left, which is great. So what I'm showing here is sort of the bottom half of my pitchfork bifurcation. So that's the black line. This is parameter. So this is taking the pitchfork bifurcation normal form. So we just have this trivial solution and then non-trivial solution. And then what we're superimposing on that is the exact solution with this linearly varying ramp in time. And there's two plots here. In the first one, we're varying the starting point. So the green, I come in here at some particular value of the parameter. And then I slowly vary the parameters in time according to that linear ramp. And I see what the exact solution predicts. And I'm changing the starting point. And this one, I'm changing the ramp speed. And so the asymptotic analysis, right, told us that I'm going to miss the bifurcation point, right? I'm going to follow the trivial solution until some point, and then I'm going to jump up and start following the non-trivial solution. And the prediction was that the jump point should be, the distance of the jump point from the bifurcation should be equal to where I started. Okay, and so we see this quite nicely here. As I start further away, I get stuck on longer. But if I keep the starting point the same and change the ramp speed, I shouldn't see sorry, I'm doing the wrong thing. That's the next picture. So this is going from the trivial solution on to the non-trivial solution. And this is going the other direction. So when we went the other direction, starting on the non-trivial solution and back, we said that there shouldn't be a jump. So we see a small sort of rounding out of the corner. And we can think of that. We haven't totally analyzed that, but it seems to be sort of a boundary layer phenomena happening there because everything's getting small. But more or less, this follows what the asymptotic analysis. So these are the exact solutions of the normal form following the asymptotic analysis. And then if epsilon gets larger, it sort of qualitatively looks the same, but we get a bigger issue happening here. So you need epsilon obviously small enough for the asymptotic analysis to make sense, but this kind of same qualitative behavior is going on. And we can push this further if we want with those exact solutions and see what's happening. And then this is the case with the varying ramp speed, right? So if we start at the same point and we just vary, so the different colors now, we're varying how fast we're passing through this. And everything, of course, is scaled by epsilon. So this is just multiplication of epsilon by differencing. So we're seeing 0.8, 1.6, 2.4. So they're all slow, but it's just how slow. And so they should all jump at the same point, and the same thing going back. So that does seem to follow what's predicted by the asymptotic analysis. But if epsilon gets larger, it's interesting. Qualitatively, we still sort of see like they start at the same point, they seem to jump down, but we start to see this sort of spreading out of the different trajectories with the different ramp speeds. And again, here the same thing. So the ramp speed, when the epsilon's not small enough, that it does seem to have an effect and sort of it causes things to spread out during the jump. So that was that's not something that we predicted from the asymptotic analysis, but we'll see that it actually shows up quite a lot in our simulations of the full system. Okay, so now we'll take a minute to think of some predictions for the full model. So the full model, once we took our model of the two neurons and we reduced it to a phase model, and we got this bifurcation diagram for the phase model with these two pitchfork bifurcations. And so if we're starting, and now we're thinking of slowly varying this GM, if I start from high, I'm thinking high to low, we'll start with that. We would expect to start on the synchronous solution, right? Prediction is there should be a delay, so a spatial delay, before we see the fact that the bifurcations occur, and then it'll jump, and then it sort of follows this. and then goes to the anti-phase solution. So that's what I've got written down here. So we expect to come along, we expect some delay, some jump, and then to move along there. And how much jump, where we see the jump will depend on sort of where we start. Okay, so now we did this with simulation of the full model. So we've got 10 ODEs, so 2 coupled of these neurons, it works out to be 10 ODEs, right? And then what I've plotted, the little circles here are the predicted bifurcation diagram based on the phase model. And then all these colored ones are looking at taking that model and applying a linear ramp to the GM current. And we start at different values. So we start some particular value and sort of close to our in phase equilibrium point. And we can see here, so I'm decreasing the GM, my solution comes along, the bifurcation point somewhere around here. I miss the bifurcation point, then I jump, and at some point I follow up. But see this, we can definitely see this rounding out here of the corner of the bifurcation there. But we definitely see the effect of the bifurcation delay sort of depending on where I start, my starting value, which is up here for my GM, that affects where my jump occurs. And if I just vary the ramp speed but start at the same point, then I more or less see that they jump at the same point. But again, we start to see the effect of this ramp speed on sort of spreading out these solutions. And again, we see this rounding off. So it's remarkably good, actually, given that we have sort of 10 ODEs and reduced to a phase model, and then we did this analysis of the phase model, and really the analysis was looking at sort of close to these, close to the bifurcation points. So it does kind of predict not too badly what's going on. And then maybe I'll quickly just go, I mean, the other way. So the interesting thing, if you go the other way, you start from low to high. Now we're going to start on this equilibrium point. We expect to see a bifurcation delay in this case, so we should miss the bifurcation jump point and jump and then follow along. So you can see already there's some hysteresis that comes depending on whether you're increasing or decreasing GM. And again, here's the bifurcation point, and we see, yes, we missed the bifurcation point, and then we jump down. Sometimes we miss it totally, and actually totally miss this whole branch of which corresponds to these other phase lock solutions. And then we come down. So this is the anti-phase going down to the in-phase. This is changing the initial GM. So this is sort of the starting point. So we see a little bit of the effect of closer, the further I start, the longer until I jump. And this is the effect of changing the ramp speed. So we start at always the same value of GM, but we change the ramp speed. And again, you see this sort of, we can call it the waterfall effect, where these different, they start to jump down, but the ramp speed is really having some effect on and how they spread out and where they go down. And it's very interesting in this case, so versus the previous case where we definitely saw these other phase lock solutions that are predicted to be stable, right? And when we decrease the GM, we definitely saw those, whereas when we're increasing, we kind of, due to the bifurcation delay, to totally miss those. And the system only sees the anti-phase and the in-phase. And just to finish up, because I think I'm just running out of time, we did some work with periodic cycling. And so as sort of expected from what I just said, you get a hysteresis loop. So if we take the blue curve here and we start, and my bifurcation's point somewhere here, we miss that bifurcation, we jump down, we totally miss what's, the other stable branch of equilibrium points, which corresponds to the other phase lock solution. So we go from anti-phase to in-phase. But then when we come back, right, so we go a little bit out here, and then when we come back, we stay on that for a long time and then jump up, and then we follow these. So whether or not the GM is increasing or decreasing makes a big difference in what we see in this sort of in-between range values. And the same thing here when we're changing, this is sort of changing the starting point and changing the ramp speed. Okay, and I think there's some time I'll skip, but this is talking a little bit about, it seemed to work better in some bifurcation points than the other, right, in terms of how well it matched. We did a number of approximations. We could control for the ramp speed and how we varied the GM, but we couldn't control on when you linearize or you do your expansion about the pitchfork bifurcation, what exactly, and we linearize those depend on the details of the model, right? And so we couldn't, if those are sort of large in some sense, that might throw off, like we're assuming all this stuff is order 1 with respect to our parameter, right? And so that's something that could throw some of this off. But let me just finish up with summarizing. So we considered this neural model with a slowly parameter GM, which changes the excitability and the synchronization property. We had looked at the fixed parameter for phase model with 2 neurons with excitatory. So here in the second half, I just looked at excitatory coupling. You can do the same thing with in phase. So with the parameter fixed, we have these 2 pitchfork bifurcations that switch from the system from in phase to anti-phase. And then we looked at the slowly varying phase model using this exact solution of pitchfork normal form and also some asymptotic analysis. And we were able with the linear ramp to sort of determine the switch point between the different behaviors and this delay in the bifurcation. And it actually was a reasonable prediction of the synchronization behavior of the full system with those sort of one-way or periodic linear ramps. So it definitely predicted these bifurcation delays and these sort of hysteresis loops that we get when we're varying this parameter. So the implication, right, if we start, go back to our original idea, we're thinking about this. If we're slowly varying this acetylcholine, right, we get this switch between synchronous and asynchronous behavior, and what the switch is, whether it's for high or low, depends on whether you have excitatory. So here's the bifurcation diagram for inhibitory. And it does this by inducing a change in excitability property of the individual neurons, right? And so one thing, the variation level of this ACA must be large enough to actually pass through the bifurcation. If it's varying slowly, right, if we saw we could delay the sort of miss the bifurcation point, if we only go to here, it might just come back again. Actually, we did see that in some cases if we didn't sort of vary it enough. So you could totally miss, right, this if it's varying slowly and you wouldn't see any change in synchronization at all. The other thing is that with the slow, if you have a slow periodic, so we increase the GM and then we decrease, then we get a hysteresis loop where we see quite different behavior depending on whether we're increasing or decreasing the acetylcholine and correspondingly increasing or decreasing the conductance on that M current. Okay, so that's all I wanted to say, and thank you very much for your attention.
That's great. Thank you very much for an excellent and very interesting talk. I'll open up the floor to questions. So if you have a question, feel free to unmute yourself or write it in the chat, raise your hand. Priscilla's unmuted herself, so would you like to ask a question, Priscilla?
Were you speaking to me?
Yeah.
Hi. And hello, Suzanne. Wonderful talk. I enjoyed it tremendously.
Hi,
and you didn't mention the word bursting, and I wonder if you're thinking of this without saying so as a bursting model.
I have to think about that. Potentially, yeah. Right now, we've just been thinking, but yeah. Yeah, it is potential, right? Where you could go from sort of a synchronized thing and then slowly into like an anti-phase type of behavior, or like so interestingly in the system, you might go from like synchronized and then have a burst of asynchrony and then a burst back into synchrony. Yeah.
Yes, and it's interesting to me. You have a jump which corresponds to the delay, which we call delay bifurcation.
Delayed bifurcation, yeah, which is like quite a common phenomenon, right? We just were kind of quantifying it.
And you have the delay going one way, but not the other way.
Yeah, that's quite interesting. It depends which way you go through the bifurcation.
Yes, and I find the same thing is true with a bursting model. If I think of your model as a bursting model, then I get this. Then I get that the asynchrony corresponds to that feature of the bursting model.
Right. So you get again this sort of hysteresis kind of loop, right?
Yes. Yes.
Yeah.
Yes. Thank you very much.
And we have a question in the chat from Alexandra. Regarding the use of GM as a bifurcation parameter, how transposable are those results, part 1 particularly, to other types of slow conductances, e.g. calcium activated currents?
Yeah, so I think, so part 1, we particularly looked at the sort of the structure of the GM current, like the way it was, where it's very simple, right? It just has an activation variable and a reversal potential. So if you had a similar structure, but if you had activation and inactivation variable, it might be a little bit more complicated to do the analysis than point 1. Although, again, people see these bifurcation diagrams like all over the place. That 2-parameter bifurcation diagram is very prevalent. So I would think it could be done with another. The second part, actually, yeah, I mean, once you sort of see that this parameter causes this effect of changing the synchronization behavior, then the sort of analysis part really didn't care much what that actual current was.
Thank you very much for that. Do we have any other questions for Sue-Ann? Jonathan Rubin has his hand up. You want to mute yourself there, Jonathan?
Yeah, sure. What happens if, honestly, I can't remember. I might have asked you this question before because I've seen you present before. So. I'll ask it again. What happens if the cells themselves are the source of the acetylcholine, right? So that the level is connected to how intensely they're spiking. Have you explored that at all?
That question, but it's a very good. So it's like then you'd have like a feedback, right?
Right.
Where yeah, no, that's a really interesting question, right? So if you then the excitability would induce, so it could be either like a negative feedback where if it gets too excitable, then it shuts off the current or something like that. No, we haven't actually thought about that, and that's really, really interesting question.
Okay.
And how it would, yeah.
Anybody else have any pressing questions? If not, I had a quick question in relation to, so you have your exact solution for the normal form, and then you have your kind of numerical solution, and you kind of qualitatively compared the two. Did you also kind of do a quantitative? Is there a way to do a quantitative comparison, or are we kind of too far?
Yes, we could. So we in the full simulations of the model, we did, at least in one direction, we could get a pretty good. So the one thing we can quantify is how much bifurcation delay we expect, right? And with the linear ramp and everything, we can quantify that we expect depending on where we start the parameter. That determines where it jumps basically. And so we did check that, and that seemed to work out at least in one direction. And that's why we're trying to figure out, because we played around with parameters, making the epsilon small enough, and it seemed to work better in one direction than the other. And yeah, I was just trying to figure that one out. But yeah, but that's the one thing that we can really quantify, right? Because that comes out of the asymptotic analysis, that prediction of how much delay you're going to get. Yeah.
Thank you very much. If there are no further questions, we'll thank Sue-Ann again. Do you want to turn your cameras on and give a little applause? And I mentioned in the chat during the talk that Mark Goldman, who missed his talk earlier today, is going to give his talk now after we'll do our official close. So I'll hand over to Massimiliano to do that. And then after we officially close the conference, Mark is going to give his talk, which will be recorded, and you can watch it back later if you like, but we encourage you to do so. So I'll hand over now to Massi, and he's going to do the thanks and closing.
Okay, so everyone, I would like to start thanking all the plenary speakers. So here we have Sue-Ann Campbell, but also the Thomas Serre and Danny Bassett for having accepted our invitation. I would also like, on the behalf of the organizer, to thank all the mini symposium organizers, so Christopher Alexandres, Louisiane Lemaire, Antonio Galvez, Suzanne Solem, Tilo Schwarga, Laura Sacerdote, and James McLaurin, who actually did the most time-consuming and burden energy-consuming work of actually approaching the potential speaker, get in touch with them, get back to us, and so on. So I think that the conference went run smoothly. The attendance was nice, higher than last year, and actually I was surprised after the 2 years of Zoom meeting fatigue. The fact that there was such a good turnout was particularly appreciated, and this is also due to the fact that the plenary speakers were particularly great. The mini symposium organizer put a very nice parallel session, and also the micro talks were stimulating and interesting. So everything was I think that everything worked nicely. I want also to thank one of the hidden somehow organizer, Willem Stannard, that actually he doesn't appear in the list of co-organizer, but he was always there at the meeting with Daniele Onie and Etienne. So he also deserved the stage, even if his name doesn't appear there. I have two more communication. The first one, or three more communication. The first one is a technical one. The streaming has been possible thanks to the fact that the Vrije University of Amsterdam and the University of Côte d'Azur gave the Zoom license to show the whole event together with the YouTube license. So thanks to them. There is the possibility of sharing your feedback about the current conference. I'm posting here, the link is available in the booklet, but I'm also posting here in the chat for your convenience. Feedback are usually, as usual, very appreciated. Because one can always, it's important to learn what went well, and especially if there is anything that can be improved. So any criticism or any constructive feedback are particularly appreciated. And actually in the feedback form, there will also be the possibility of letting us know your view for next year. So for those of you who weren't there yesterday, or even if you were there was a discussion about the future of ICMNS. Okay, so at the moment, the current position is, which is actually a possibility, is to hold a meeting next year, hopefully in person, jointly with CNS. So the idea would be to run the conference at some point around mid-July, ICMNS and CNS. So Computational Neuroscience and International Conference on Mathematical Neuroscience together at some point last year. Nothing has been decided. That's a possibility. So your view on this, your feedback on this is particularly appreciated. There will be further communication. There will be an email coming with the minutes from yesterday meeting. So that will give you a broader overview. But for the time being, you might start thinking whether, the possibility of having a joint meeting next year is something that you find appealing. And if you see any possible criticism or doubt, we will appreciate if you can list them. And yeah, if you want to get involved in any way with the organization, that's also something that you can mention in the form, or you might directly approach us. And I think that I cover all points, hopefully. Yes? Yeah, I was talking quite fast. I need to go and pick up my daughter, otherwise she got locked in the nursery. But hopefully I see that Daniele Onie are saying yes, and also Etienne and Willem. So everything seems to be fine. if there is any other comment, or if you want to add anything else, just feel free to unmute yourself and jump in.
I think in the interest of time, we should try and leave the floor to the one last talk that we have to go through before the end of the conference.
Okay, so as I said, the form is there in the chat. Feel free to approach us anytime. And then I'm sorry to have to miss Mark Altman talk, but I think that the floor is yours now. Mark?
All right.
Okay.
Can you hear me?
Yes.
Great. And do you see my slides in slide sorter?
Yes, we see full screen with also the cursors there at the moment.
All right. Well, thank you very much, and apologies on the time zone mix up there. But very happy to be here and excited to talk about some work. This is from the Balanced Networks track, and I want to talk about balanced networks for working memory and neural integration. Just to introduce things. In many memory and decision-making circuits of the brain, we see an accumulation that is a mathematical integration in the sense of calculus and maintenance of signals for on the time scale of 1 to 10s of seconds. So here's a cartoon version of that. Stimulus is shown in yellow. While the stimulus is present, neuronal activity is accumulating, mathematically integrating that input. And then in the offset of the stimulus, the value of that accumulation or the value of the stimulus is stored in working memory. And I want to note that this can be an analog storage corresponding to the accumulation value during the accumulation period. Now, this has been a long-standing puzzle in the theoretical neuroscience and community, and for a very simple reason, that most neurons intrinsically have very brief memories. That is, if you present an input stimulus and turn it off, typically the neuronal or synaptic time constants are in the orders of tens to hundreds of milliseconds, not the 1 to 10 seconds of working memory. So the big question for today is, can cortical-like balanced networks generate persistent neural activity and perform neural integration? I just want to sort of set us back to the beginning in looking at classic balanced networks. So this is from the original groundbreaking paper of Carl von Vrieswijk and Chaim Sampolinsky. And it had the following sort of classic excitatory, inhibitory, recurrent cortical architecture. And when you looked at an individual neuron in those networks, well, there was a certain amount of excitation which fluctuated over time, and a certain amount of inhibition shown here in the bottom fluctuating over time. But the net input to any current was close to zero. And in particular, it was fluctuations of that near zero net current that caused spiking, and that led to the classic irregular firing that's a hallmark of cortical neural activity. But from the point of view of talking about slow timescale persistent activity, these networks, they went on to show, have a very fast response. They said an advantage of them is that they could very quickly and closely follow external inputs. But this means if an external input turns off, these networks would quickly turn off. That is to say, they're not good for memory storage. So instead, going to other classic work, memory models haven't been based on balanced networks. They've been based on positive feedback. And this is overwhelming across many, many different models. When you distill them down to their essence, they come down to something like I'll show on this slide. So the idea here is that an input coming into a neuron, if it comes in a typical isolated neuron, its firing rate will go up and then come down, quickly decaying on some neuronal or synaptic timescale. But of course, neurons aren't in isolation. And the point is that when they're not in isolation, they're connected in a network, they can now have persistent activity and respond to a command input. And if there's a second input, they can furthermore increase their level of persistent activity, that is, temporally integrate the input. How does this occur mechanistically? Well, the story is this command input, comes in. If the neuron were in isolation, the activity would decay away, but there's positive feedback. So there's positive feedback through recurrent excitation that can sustain the activity. And there's also another form of positive feedback through a double negative form of positive feedback or recurrent disinhibition. And when this is tuned properly, this feedback can exactly offset the natural leak in the neurons so that basically activity can be maintained as the feedback is canceling the leak. And if a new input comes in, the input can again drive the neuron to increase its firing rate and hold that increased firing rate. So this is an analog form of persistent activity in the absence of input. Okay, but what's the problem with these networks? Well, now let's look at the two different neurons. We see neuron 1 here stepping up its activity. Of course, the recurrent inhibition means that the other neuronal population, population 2, decreases its activity when these commands come in. And what this means is it has strong implications for thinking about the balance or lack of balance of synaptic currents coming into any given neuron. So now let's look at up here, these inputs coming into neuron 1. So we have excitatory inputs highlighted in green, inhibitory inputs highlighted in red. And now let's think about what happens as neuron 1 increases its activity. So we're plotting here on the y-axis, the inhibitory input to neuron 1, on the x-axis, the excitatory input to neuron 1. And now here's neuron 1's firing rate. As neuron 1's firing rate increases, shown in a diagonal, each of the points here indicates the amount of excitatory and inhibitory input. And you can see that these are imbalanced. At 30 Hertz, there is one value at which there can be a balance, then inhibition and excitation is exactly balanced. But as the neuron increases its firing rate, it gets more excitation and less inhibition, and vice versa. If its firing rate decreases, it has reduced excitation and more inhibition. So this shows that the classic positive feedback models are not balanced in general, or they are balanced at only a single firing rate. So just to make really clear, this does not look like cortical networks. And this is some work from originally David McCormick's lab showing during persistent activity, the inhibitory conductance and excitatory conductance coming into various neurons. And what you see is this classic positive correlation of the inhibitory and excitatory conductances. In other words, they're moving around something similar to a 45-degree line, with some fluctuations. But the point is when inhibition goes up, excitation goes up. And that is exactly opposite what the positive feedback models look like. Okay, so what this suggests is maybe we need a different principle. And in work a number of years ago with Sukbin Lim, we actually looked at a different framework for thinking about how persistent activity can be maintained. And it came from control theory. And it came from a sort of basic result in control theory that strong negative feedback of a signal produces an output equal to the inverse of the negative feedback signal. That is to say, if there's an input X, which goes through some transformation, can be linear or nonlinear, to produce an output Y, and then the output Y is fed back very strongly through some transformation, in this case a derivative, then the output Y will be proportional to the inverse of that transformation, and the inverse of a derivative, of course, is an integral, so that you'll get that Y is proportional to the integral of X. Let's translate that to a memory circuit and get what the essence is. So here is the basic cartoon. We have a memory cell, and it's going to receive negative derivative feedback. Well, what does negative derivative mean? It's really a slip detector. Every time the memory starts to decay or grow, it's being opposed by this slip detector, and that's being fed back with some strength W derivative. So now if we take our garden variety simple model of a neuron, linear model, tau drdt equals minus R plus its inputs, Now we have a negative derivative feedback input. And you can very quickly see that a negative derivative feedback on the right is the same as mathematically equivalent to just increasing the time constant of the neuron. So this is how we can get very slow persistent activity, is basically the strength of the derivative feedback corresponds to the time constant of the neuronal decay. A more physical picture is that having a term which is proportional and opposite to the derivative is like viscous drag. So it's like having it in terms of the attractor formalism for memory, it's like putting molasses or a very viscous substance so that the state of the system decays very, very slowly. It does still decay, but very, very slowly in a way that allows for long time scale persistent activity. And that means basically, again, if the activity were going to decay, there's a positive corrective signal opposing that decay, or if it were going to grow, there's a negative corrective signal opposing that growth. Okay, so now the question is, how can this play out in neural circuitry? How can we build a derivative in the neural circuitry? And a very, very simple architecture does this, and that's shown right here. So here we see a neuron with just a firing rate neuron characterized by its rate R of t. And imagine that we have a slow positive feedback pathway shown here and a fast negative feedback pathway shown here. And just to remind you, just very trivially, that negative of a derivative goes as R of t minus dt minus R of t. That is, putting it into the context of firing rates, we need to have feedback that has equal amplitude, coefficients 1, but offset in time to perform the derivative operation. So negative derivative feedback naturally arises when positive feedback is slower than negative feedback, and when the amplitudes of these are equal, so excitation and inhibition are balanced. And one can just carry through the math, and it's in this paper. But if you label the strengths of these connections as J and refer to the positive feedback pathway as having a time constant tau plus and the negative feedback pathway as having a time constant tau minus, then one gets that the time constant for the network goes as W derivative, and that goes as J, the strength of the input, times the difference between these time constants. And this immediately shows why the classic work did not produce this slow, persistent activity. The classic von Vrieswijk and Sampolinsky work had the same time constant on each of their pathways. So we didn't have this derivative feedback effect. Okay, well, this is a very toy model violating Dale's principle. So how do we flesh this out into a more cortical-like network? Well, we have one which again looks like the classic von Vrieswijk Sampolinsky one, except for the time constants. We have an excitatory pool, an inhibitory pool, and here we have the slow positive feedback pathway. And if this inhibitory loop is fast, then we have a fast inhibitory feedback pathway. And again, if those pathways are balanced in strength, but with these appropriate time constants, we get derivative feedback. Okay, so let's check that this actually works and maintains persistent activity and performs neural integration. And that's shown here. Indeed, it does. So this is showing response to two different types of inputs. On the left, you see the response to pulse inputs of 3 different amplitudes. And you can see that the firing rate, again, maintains persistent activity as very slow decay. And that's due to the slip correction of the derivative feedback. In response to steps, this shows that it really is a mathematical integrator. In response to 3 different levels of step inputs, you can see 3 different slopes of accumulation, of ramping activity. So it really is an analog memory network which can temporally integrate its inputs. So are there interesting properties about this from this network that you don't get in the positive feedback networks? And that's shown, and indeed there are, and that's shown here. So what I'm going to show you here is the effect of perturbations on the network. And one of the things that's well known is that these networks, which are mathematically line attractors, are very not robust. And typically for the positive feedback networks, they're not robust to almost any perturbation one can imagine. So here's just the cartoon of a simple excitatory positive feedback network. One could also do the double inhibitory one. But if one kills some fraction of the excitatory cells, or in the double inhibitory one, kills some of the inhibitory cells, or changes gains, or scales excitatory inhibitory synapses, or puts any nonlinearities in, the network detunes. And that's because basically any of these manipulations will remove or disrupt the positive feedback. So this is showing in green either +2 or -2% changes in the feedback level or in the number of cells. And you see already dramatic loss of the persistent activity. Now in the middle row, look at the derivative feedback network. Now imagine killing some fraction of these excitatory cells. Well, killing those cells doesn't disrupt the time constants in the network, so it doesn't disrupt that positive feedback is slower than negative feedback. And likewise, in terms of the strengths of the inputs, killing some of the excitatory cells, well, it reduces the excitatory feedback pathway, the positive feedback pathway, but it also equally diminishes the negative feedback pathway, so things still stay in balance. And therefore, here showing 10% increases or decreases in whatever of these perturbations, killing cells, increasing gains, etc., hardly changes the activity at all. And can one have a combination, kind of have one's cake and eat it too? Yes, one can. So by adding a little bit more excitatory feedback, one can have a net overall positive feedback, and then one can have perfect persistent activity and also this robustness to perturbations. Okay. So another property I said of balanced networks is that they have irregular spiking. So we also built a spiking model with populations of excitatory inhibitory neurons. And this is showing just one of the many levels of persistent activity it can maintain. And you can see the very irregular spiking. And if you actually calculate the CVs of these the spike trains of different neurons, you get a distribution across neurons. This was actually like really a mean field type model where everything was just randomly connected. So it was balanced, it was loosely bound, it was balanced on average in a mean field sense. But you see this distribution of coefficients of variation in the interspike intervals looks very much like what people see in working memory experiments. Again, high, CVs of 1 or between 1 and 2. All right, so to compare this to the positive feedback network that I showed you, well, that one was one where there was competition between two populations. And here we set up a competing EI networks, if you wish. So here's this, again, this motif of the E and the I, and again, it's gonna have slow positive feedback and fast negative feedback, but now they compete because the excitatory neurons from one pool excite the inhibitory neurons of the other pool, and any mutual excitation, it can be removed entirely, but it was weak. One gets out of this persistent activity. Again, here's population E1 and population E2, where one population goes up when the other population goes down in response to an external input. Again, it's robust against changes in synaptic weights. But importantly, what I want to show you is, unlike the positive feedback motif, now if we look at the inputs, let's say onto neuron 1, the excitatory inputs and the inhibitory inputs, that's what we're plotting here. The inhibitory inputs to E1 on the y-axis, the excitatory inputs to E1 on the x-axis. Now you can see that as E1 moves through different firing rates, these inputs remain balanced. So we have a balance of excitatory input throughout the firing range, which looks more like what we think of as the experimental motif of increased balanced excitation and increasing excitation and inhibition together in cortical networks. Okay, so from the last little bit of this talk, I'd like to actually switch to not just talking about just balanced networks for storing one memory, but talking about spatial patterns of input. So this is a classic experiment from Funahashi and Goldman Rikic. A monkey was asked to fixate, and then a light came on at one of 8 locations in a circle. When the light went off, the monkey had to make a saccade, and different neurons were tuned to different locations. So this is showing the activity during the D is the delay period for different neurons. And this neuron shown here was tuned to angles between 90 and 180 degrees, Q locations between those, and didn't respond to other Q locations. And the question is, how can these patterns of neural activity corresponding to this type of behavior be carried out? And there's a very classic architecture for these spatial working memory tasks. It's known as the ring model. There's a ring of neurons. They're connected locally by excitation and more distally by inhibition. And these do a great job of by symmetry. They can encode any location of a spatial input, but they don't encode amplitudes of the inputs. So this is shown on the right. So if the input is small, there's no activation in the network. If the input is 0.1 or even 5 times that, 0.5, you get exactly the same activity during the memory period. It's an all or none response, and there's no encoding of input amplitude. And the question I'm going to ask and what I'm going to show is, can a balanced ring model encode amplitude information? So the architecture now is going to be similar to what I've shown you before. There's going to be this core of this EI motif with slow positive feedback and faster negative feedback. And then I've unrolled the periodic connectivity. But what you can see is, again, there's going to be lateral inhibition and lateral excitation defined by weight matrices of the populations JEE, JII, JEI, JIE with appropriate time constants to have slow positive feedback and fast negative feedback. So what's the key principle here to make a balanced network in these spatially extended networks? Well, the key principle is to use Fourier analysis. So again, if you're going to see a firing rate pattern that's sustained like the following one, you could break it into its Fourier components. There's a DC component, there's this component, first cosine component, a second cosine component. And the idea is any component of activity that can be maintained is maintained by derivative feedback on that component. That, of course, leads to the question, how do you form this derivative feedback on the components? Well, without getting into the details, the way you realize this is you do a Fourier decomposition on the weight matrices, so that for any component of the weight matrix, you can break it into its Fourier components. And now we have the same balanced network that I showed previously, but the tuning that needs to be done is there needs to be, again, slow positive feedback, fast negative feedback with a balance of the strengths of the connectivity within any Fourier mode. When you do this, and I should make clear that even though it uses a Fourier decomposition, it still works with nonlinear neurons. So that's a nice feature of it. You can now maintain memory activity for different amplitudes of inputs. So what's being shown here was four different trials with 4 different levels of input from orange up to blue. And you can see that the amplitude of the input is now stored in the persistent activity pattern. So the bump of activity can now encode amplitude information. And what I'm not showing you for the sake of time is that the network will also temporally integrate inputs at a given spatial location. And it's also robust to either local or global perturbations of cells in a way analogous to the network I showed you earlier. So to summarize, what I've shown you is that classic balanced networks have fast dynamics, and classic working memory and neural integrator networks are based on positive feedback and are not balanced. What I've shown you instead, if one wants to make a slow dynamics network that maintains slow persistent activity and performs neural integration, that can be done through negative derivative feedback networks that are based on the principle of balance, excitation, and inhibition, but now with slower positive than negative feedback. These are robust to many common perturbations, such as death of cells, and the properties extend to nonlinear spatial memory networks by Fourier analysis. Thank you.
Thanks so much, Mark. Great to hear. Are there any questions?
Rainer?
I have a couple of questions. So with the spatial networks at the end, so you have, I hope you understand this, but you have spatially varying connectivity strengths.
Yes, so the connectivity strength is spatially varying. So the simplest thing you could do, which would be similar to the original ring models, would just be to tune the first cosine component of the connectivity, and then what you get is a network which always holds a bump with a cosine shape. But the key thing is, again, when you break down the spatial connectivity around the ring into its Fourier decomposition, then you're going to have the excitatory to excitatory feedback it's actually shown here. So the excitatory to excitatory feedback in that mode has to equal the negative feedback, which would be JIE, that's going around the loop, and then it's normalized by the JII term. So that's the condition right there, and equality between those maintains the balance, and you do that within the Fourier modes.
Right. Is that a role? How do you do that computation? Does it get very complicated, or it's just?
Yeah, computationally, you literally just say, I want this connectivity, and then you just do a Fourier analysis. You just stitch together the sines and cosines.
I see. Yep. Oh, very interesting.
And then you end up with the overall connectivity for the network.
I see. Yeah, very interesting. I was also wondering, so what parts of the brain exactly were you saying are these balanced networks present, like sort of all over, or any parts in particular?
Yeah, so I would love someone to do the experiment. No one's actually done the intracellular recording experiment in vivo during an analog working memory task or a neural integration task, which also requires, of course, not just doing the experiment, but first localizing where that activity is maintained, rather than possibly just looking at a readout, which wouldn't have to be balanced. I think, but one thing would think of this as something like in the frontal cortices.
Okay.
And at least I think it may be the wrong layer of neurons, but there were some studies showing that, for example, the NMDA receptors in the E to E connections are slower than the NMDA receptor kinetics in the E to I connections. So that would actually be the right way to have this, the right way to have the kinetics, at least the slower positive feedback, the negative feedback connectivity. For the balance, it needs to be shown experimentally, but the idea would be, given one sees irregular firing, and at least in, I believe the McCormick work may have been either slicer and anesthetized work, at least their excitation and inhibition go up and down together. This is what one would expect.
Mm-hmm. Okay, interesting. We have a question in the chat you might see from Ganendra Singh. He asks, how much is it different from the Cowan model with feedback mechanisms?
Yeah, so I mean, this is the basic Wilson-Cowan model. The real key thing in here is, this is both similar to the Wilson and Cowan. It's also similar to the, von Vrieswijk, Sampolinsky architecture. The real key thing was one simple change. And the one simple change is just to make this synapse right here slow. This JEE synapse slow. It actually also helped. The positive feedback also includes the JII if you work through the math, and you can think of inhibition is like positive feedback. So the overall positive feedback time constant is tau EE plus tau II, and that needs to be greater than the negative feedback pathway, which is going around this loop, tau EI plus tau IE. But so that's the key thing, is changing the time constants.
Okay, thanks, Mark. There's another question here from Jung Young Kim. Can you see it in the chat? Okay, so you're saying,
right, so
you can speak.
Scroll down, yes. Yeah, can you still have a nonlinear neuron and have it apply to the neural integration? So that, so I showed actually in, I guess, the next slide after this one, that actually this neuron is nonlinear. So you can, and the nice thing about, and the way to think about that nonlinearity is the input-output function of the neuron being nonlinear doesn't really cause a problem, because you're not disrupting that it receives that the inputs coming into the neuron are balanced, and you're not messing up the relative time constants of the excitatory and inhibitory feedback pathways. So the nonlinearity doesn't disrupt that. Now, will it strictly, with this output nonlinearity, will it strictly be a mathematical integral? It does look like it linear. I think it still does linearly integrate. And again, the key thing goes back to this control theory result, that if you feed back a derivative very strongly, it overrides the intrinsic properties of the network. And then you get a very linear response. The same thing occurs, actually, Bart Ehrmantraut wrote a paper years ago talking about how you get linear input, linear firing rate versus current injection relationships in conductance-based neurons. And again, it was a very similar principle of having very strong negative feedback overriding the other intrinsic channel properties so that those details didn't matter so much. So I think that, so the basic principle is that you're not messing up the derivative feedback and you're not messing up the balance of inputs.
Okay, well, thanks so much, Mark and everyone else. I think we'd better call it a day because we're a bit past the time, but it's great that we could catch your talk at the end, and it's a very interesting work. So thanks again.
Thank you very much. And thanks for your flexibility.
No, thanks for making it. It's great. Okay, so I'm told that I'm meant to just formally close the meeting now. So
thank you very much, James, and everyone.
Yeah, thanks, Danielle. Thanks, Danielle and Etienne and Anya for organizing. It's great.
And James, thanks so much, and apologies about the mix-up. I don't know if I've used